Hello everyone, my name is Vikas Authan and welcome to the today's session of paper discussion for the NEET 2024. So as we have analyzed the paper, the complete biology part have came from the NCERT, maximum questions have came from the NCERT part. So I am Vikas Authan and today I will start from the botany section. The question wise analysis we will do and solve the questions in front of you. Let's start the first question from the botany part. So from section A, we are going with the question number 101. Identify the set of correct statements. So from here, we have to see which statements are correct. First of all, the flowers of Vallisneria are colorful and they produce nectar. Actually, Vallisneria is an aquatic plant. So usually in the aquatic plants, the colorful flowers are absent. So this statement is incorrect. Okay. So if A is incorrect, we can say that option 1 and option 4 are automatically incorrect here. Then flowers of water lily are not pollinated by water. Yes, this statement is true. Water lily is actually growing in water, but it pollinates by the wind as well as by the insects. So this statement is true. Third, in most of water pollinated species, the pollen grains are protected from the wetting. Yes, this statement is true. From NCERT, they have given that the pollen grains are protected by a mucilaginous covering. So this statement is true here. Pollen grains of some hydrophytes are long and ribbon-like. Yes, this statement is also true. And finally, in some hydrophytes, the pollen grains are carried passively inside the water. So what happens in the water? The pollen grains are flowing passively. So this statement is also true. So according to this, we can say that option number 2, that is statement B, C, D and E are correct. Okay. So this is a very easy question from the sexual reproduction in flowering plant. Now let's come to the second question. In the type of conservation in which the threatened species are taken out from their natural habitat and placed in a special setting where they can be protected and given special care is called. Now here, although this question is not from directly from the NCERT statement, but few options we can eliminate here. Let's see. First of all, they are saying the word semi-conservative method. Semi-conservative method we have studied in the DNA replication. So this statement is not correct here. Second, sustainable development. This is also not related to the conservation method. Third, in situ conservation. Actually, this method is not of in situ, it is a ex situ conservation, and ex situ conservation comes under the type of biodiversity conservation. So exactly, they have not used the word in situ conservation, but broadly we can say this, it is a type of biodiversity conservation. Now let's come to the next question. Inhibition of succinic dehydrogenase enzyme by malonate is a classical example of. So actually in NCERT there is a topic given that competitive inhibition. So suppose this is an enzyme, here we are representing an enzyme. Okay, now in enzyme there is an active site, this is an active site, here a substrate binds to this active site. So now what happens in competitive inhibition, there is a molecule which is called inhibitor, and this inhibitor is structurally analogous to the substrate. So this inhibitor, if it binds to the active site, it can inhibit the activity of enzyme. So basically what I'm trying to say here, in competitive inhibition, substrate and inhibitor, both of them compete for the same active site. So here in NCERT it is given that for the succinic dehydrogenase enzyme, there is a we can say malonate is used for the competitive inhibition. So malonate is an example here which is used as an inhibitor. Okay. Now next question. So this is a diagram based question. Identify the part of seed from the given figure which is destined to form the root. So actually if you see in an embryo, okay, in an embryo the radical part develops into the root. So it is a transverse section of a monocot seed, representing the option C as radical here. Option C is represented as radical here, and this radical forms the root in these seeds. So answer will be option number 1, that is C. So C is representing the radical here. Okay. So this was a diagrammatical based question exactly coming from the NCERT. Next one. Bulliform cells are responsible for. So exactly what happens in the bulliform cell? Now, in the case of monocot plants, whenever there is a water stress, Water stress. Water stress means if there is a low availability of water. So under the low availability of water, what happens? These bulliform cells, they shrink, and what they cause? They cause the we can say inward curling of the leaves. What is inward curling? This is the inward curling of leaves. Okay. Now why this happen? This happen under the water stress. Water stress means if in a soil water content is low. Second thing, if in a soil water content is low, what will happen? The transpiration can be more. So to reduce the transpiration, the leaves, they undergo curling. So answer will be option number 3. Okay. Now next question. Which of the following are required for the dark reaction? Okay. So as we know that for the photosynthesis, light reaction and dark reactions are required. The key products of light reactions are NADPH2, oxygen and ATP. Okay. Now among these, during the dark reaction, two major products are required. One of them is ATP and second is NADPH2. Third thing, actually in dark reaction, CO2 is undergoing the process of reduction. That is, CO2 is undergoing the fixation. So there are three main requirements for the dark reaction. One is the ATP, okay, one is NADPH and one is CO2. Means C, D and E statement are true. So answer will be option number 1, C, D and E. Now let's come to the next question. Formation of interfascicular cambium from fully developed parenchyma cell is an example for. So what is a interfascicular cambium? So interfascicular cambium, as we know, it is we can say it is developed during the secondary growth in the plant. It is formed during the secondary growth in the plant, and it is a type of we can say secondary tissue, or we can say it is a type of secondary tissue which grow in the plant during the secondary growth. So basically, whenever a permanent tissue or permanent cell which have lost the ability to divide, if it regains the ability to divide, then this process is as de-differentiation. We call it de-differentiation. Okay, what is differentiation? Differentiation means the cells have become matured and they have lost the ability to divide. Now what is de-differentiation? De-differentiation means removal of differentiation. So de-differentiation is a process seen here. So formation of interfascicular cambium from a fully developed parenchyma cells. So here parenchyma cells are what? They are mature cells. They are differentiated cells. And when they undergo de-differentiation, they will form the interfascicular cambium. Now next question. HIN2 always cuts the DNA molecule at a particular point called recognition sequence, and it consists of. So basically there is a point called the recognition sequence. So what is recognition sequence? Basically in a DNA there is a sequence of base pair in which a restriction enzymes makes and cut. Usually these recognition sequences are made up of 6 base pairs. They are made up of 6 base pairs. So answer should be option number 4th, that is 6 base pair. Okay, next question. Tropical regions show greatest level of species richness. Why? What is the reason? Let's understand this point. First of all, tropical latitudes have remained relatively undisturbed for millions of years. Hence, more time was available for species diversification. So what happened in the tropical region? For many years, they have left undisturbed. So species have undergone the diversification. Yes, this could be a reason. Second, tropical environments are more seasonal. This statement is incorrect. Actually, tropical environments are less seasonal. Less seasonal. This statement have been taken from NCERT. Third one, more solar energy is available in the tropics. Yes, this statement is true because our Earth, this is our Earth and this is the equator. Okay, tropical region lies near the equator, and in the equator maximum sunlight is incident. Maximum sunlight is incident here. That's why we can say the maximum energy is available. Fourth one, constant environments promote niche specialization. Yes, if the environment is constant for many years, the species or the we can say organism, they undergo specialization where they do the ecological function. So what is niche? Niche is a area in a we can say habitat where an organism show the ecological function. So yes, constant environment promote the niche specialization. Fourth, tropical environments are constant and predictable. So we can say that yes, the tropical environments are more predictable because we can say here they have remained unchanged for many years. So according to this, I can say only B statement is incorrect. So answer will be A, C, D and E. Coming to the next question, which one of the following is not a criteria for the classification of fungi? So usually you have studied that fungi are classified into 4 main classes. Okay, starting from the we can say Phycomycetes, then Ascomycetes, Basidiomycetes and Deuteromycetes. So this classification is mainly based on mode of spore formation, morphology of mycelium and the fruiting body. It is not based on the mode of nutrition. So answer will be option number 4. Now, how many molecules of ATP and NADPH are required for every molecule of CO2 fixed? Every molecule means they are asking for one CO2 molecule. So one CO2 molecule, how many ATP and NADPH2 are formed? So as we know, during dark reaction, there are 3 main steps. First step is called carboxylation. Okay. Second step is called reduction. And third step is called regeneration. Okay. Now, in carboxylation, no ATP or NADPH2 are utilized. Second, during reduction you will say that 2 ATP and 2 molecules of NADPH2 are utilized, and during regeneration 1 ATP is utilized. So total how many ATP will be there? There will be 3 ATP and NADPH2 will be 2. So we will say that according to this 3 molecules of ATP and 2 molecules of NADPH are required during the dark reaction. Okay, next question. These are regarded as major causes of biodiversity loss. So in NCERT you must have heard about the evil quartet. Evil quartet is what? Evil quartet means there are 4 main factors which are responsible for the biodiversity loss, starting from first, that is overexploitation. Yes, overexploitation is responsible. Second is co-extinction and habitat loss and fragmentation. So these 3 factors are responsible here. So I can say answer should be A, B and D, that is option number 2, A, B and D. Okay, next question. The capacity to generate a whole plant from any cell of the plant is called. So this property is called totipotency. So what is totipotency? Toti means total. Okay, and potency means ability to generate a complete new plant. Okay, it is the ability to generate a complete new organism. Okay, so totipotency will be the answer for the capacity to generate a whole plant. Okay, coming to the next question. The equation of Wohlhurst-Per logistic growth is, this is the equation they have given, dN by dt equals to Rn, k minus n by k. They are asking what does we can say k represent here. What does k represent here? They are asking in the question. So k is representing the carrying capacity. It is representing the carrying capacity. Next question. Spindle fibers attached to kinetochores of chromosome during. So exact from the NCERT they have given this, that we can say in a chromosome this is the centromere and these are the chromatids. On chromatids we have 2 disc-like structures which are called kinetochores, and on these kinetochores the spindle fibers gets attached. So this happens during the metaphase of the cell cycle. Now we will come to our next faculty, that is Shiny ma'am. She will continue from this. So let's close this file. Okay, so coming to the Shiny ma'am, I'm forwarding to her. Just keep the mic. Ma'am, take the mic.
Should be like this one. So hello students, I hope your exam went well. Now let's see the next set of questions. Your question number 116 is identify the type of flowers based on the position of calyx, corolla and dendrotium with respect to the ovary from the given figure A and B. Now if you have studied NCERT very carefully, these are your NCERT diagrams only. In your NCERT, in your position of ovary, we have 3 diagrams or basically 4 diagrams: hypogynous ovary, perigynous ovary and your epigynous ovary. Now these 2 diagrams are of your B and C diagram from the NCERT, which are your perigynous ovary. So the correct answer is your second, A perigynous and B perigynous. Now let's move on to the next question. Match the list 1 with list 2. So the list 1 have your Rhizopus, Astilago, Puccinia and Agaricus. These are your types of fungi and we have their common names in the list 2, that is mushroom, smut fungi, bread fungus and your rust. Now choose the correct answer from the given options. So the first one is Rhizopus. Rhizopus is your bread mold. Then Astilago forms smut, and then Puccinia, it is the rust fungus, and then Agaricus, very commonly we know it is the mushroom. So the correct answer is your first option. Okay. Now let's move on to the next question. Okay, next question is, in a plant, black seed color BB and capital B small b is dominant over white seed color small b small b. In order to find out the genotype of the black seed plant, with which of the following genotype you will cross it? Now the question is saying we have this represents your black seed and small b represents your white seed. Now the question asks, which of the following genotype, which genotype should we cross to get black seeds? So which genotype should we use to cross so that we can get the black seeds? Now this is what? We have to find out the genotype. This is a type of test cross. So in test cross what we do? We cross the F1 with our recessive parent. That's what we are going to do here. We are going to cross the BB or the capital B with your small b. So the correct answer will be fourth one. Now the next question is, a pink colored snapdragon plant was crossed with a red flowered snapdragon plant. So the genotype of pink snapdragon is capital R and small r, we have studied in NCERT, right? And for your red flowered snapdragon, the genotype is small r. The capital R and capital R. Now when we cross this, now the question says, what type of phenotype is or are expected in the progeny? Now when we will cross it, we will get capital R, capital R, capital R, small r, capital R and capital R, small r. 50% progeny will produce our red flowers and the other 50% will produce the pink flowers. Hence, the fourth option will be correct, that is red flowers as well as pink flowered plants. Okay. Now let's move on to the next question. Next question says match the list 1 with list 2. So the list 1 have a 2 or more alternate form of genes. The alternate form of genes are your alleles, right? Next is cross of F1 progeny with homozygous recessive parent. Now cross with F1 progeny with homozygous recessive parent we just saw in the previous question, which is your test cross. Okay. Next, C is cross of F1 progeny with any of the parent. Of course, that is your back cross. And the left is number of chromosome sets in plant. It represents the ploidy. So the correct option is first one. Okay. Now next question is lecithin, a small molecular weight organic compound found in living tissues, is an example of. Now if you have studied cell membrane, there we study the phospholipids. So what is lecithin? Lecithin is a type of phospholipid which is abundantly present in your cell membrane. So the correct answer is fourth, phospholipids. Now the next question we have is match list 1 with list 2. List 1 represents your different types of fungi and the products which are formed. The list 1A is Clostridium butyricum, which forms your butyric acid. Then second B is Saccharomyces cerevisiae, which forms ethanol. And then C is Trichoderma polysporum, which forms your cyclosporin A. And then D, the last one is Streptococcus species, which forms streptokinase. So we have A third, B first, C fourth and D second. First option is correct. Now let's move on to the next question. The next question says, in the given figure, which component has thin outer walls and highly thickened inner walls? So in the stomata we have studied, we have guard cells. And these guard cells have a thin outer wall and a very thick inner wall present in them. So here, which A, B, C, D, out of which one represents your guard cells? Your C part. Okay. D represents your stomatal aperture. So the correct answer is C. Okay. Then next question number 124. Which of the following is an example of actinomorphic flowers? The actinomorphic flowers are those which represents the radial symmetry, right? The radial symmetry is seen in out of first Pisum and your Sesbania belongs to the family Fabaceae, and they will represent bilateral symmetry. Or you can say zygomorphic symmetry. The case here shows irregular symmetry. We have only one answer left, that is Datura belongs to Solanaceae family, which represents actinomorphic symmetry. Okay. Then the next question. Question number 125. A transcription unit in DNA is defined primarily by the three regions of DNA, and these are with respect to the upstream and the downstream end. So now we have to tell about the transcription unit. The transcription unit of your DNA is divided into three parts. The first one is your promoter sequence. Then we have the structural gene. And then at the end we have the terminator or the terminator sequence. Okay, so the correct answer for your transcription unit is second, promoter, structural gene and terminator gene. Okay, then next is, what is the fate of a piece of DNA carrying on gene of interest which is transferred into an alien organism? The DNA which is we have inserted in an organism, what is the fate of it? What happens to it? Okay, so the first one is the piece of DNA would be able to multiply. It is multiply itself independently in the progeny cells. Is it correct? No, it won't be able to multiply itself independently. Second, it may get integrated in the genome to the recipient. Yes, this is correct. Then C, it may multiply and be inherited along with the host DNA. Yes, it will multiply, and yes, it will get inherited along with the host DNA. And then the D option says the alien species of DNA is not an integral hoga beta, integral part of chromosome, which is also incorrect. Then the E part shows, it shows ability to replicate, which is also incorrect. So the correct answers from the given option below is B and C only. Now the next question, 127. Oxen is used by gardeners to prepare weed-free lawns, but no damage is caused to grass as oxen. Why oxen does not do any damage to grass? What is special in grass? Can anyone tell? Yes, you know that grass is a monocot and oxen does not affect your monocotyledonous plant. So the correct answer will be first. Then 128th question, the cofactor of the enzyme carboxypeptidase is, so the cofactor which enzyme carboxypeptidase use is your zinc. Okay. Then next 129 question, the lactose present in the growth medium of bacteria is transported to the cell by the action of. Now you know that lactose will be, we have three types of gene, permease, polymerase is your, polymerase, let's delete that, it's not even related to this topic. We have permease, beta-galactosidase and acetylase, which helps in transport of your lactose is permease. It helps the lactose to transport in the cell membrane. Now we have next question, that is which one of the following can be expressed on the basis of Mendel's law of dominance? Out of these 5 statements, which can be explained in Mendel's law of dominance? First is, out of one pair is dominant, out of one pair of factors, one is dominant and the other is recessive. Yes, this is correct. Second is, alleles do not show any expression and both the characters appear as such in F2 generation. It is incorrect. Alleles do show expressions and both of the characters do appear in F2 generation. Do not is incorrect here. Now see, factors occurs in pair in a normal diploid plant. Yes, this is correct. And then D, the discrete unit controlling a particular character is called factor. Yes, this is the line from NCERT only. And then E is, the expression of only one of the parental character is found in monohybrid cross. Yes, in monohybrid cross, we only take reference of just one character. Okay, hence how many statements are correct? A, C, D and your E. Second option will be correct. Sorry. Fourth option is your correct, A, C, D and E. Now let's move on to the next section, which will be taken by Sangeeta ma'am.
We have to hold or we have to keep? It will look odd now. Okay. It's Hello students, we are going to continue with the next part of the questions. Okay, so coming to the question number 131. So this is the question from biotechnology. The Bt toxins are insect specific, so and coded by a gene called Cry1AC. So actually we are going with 3 genes. For example, Cry1AC and Cry1AB, Cry2AB. These 2 genes are for cotton bollworms and tobacco budworms. And Cry1AB alone. The Cry1AB alone will be for corn borer, right? So Cry1AC and Cry2AB is for cotton bollworms and tobacco budworms, and Cry1AB is for corn borer. In the first statement, if you see, the Bt toxin are insect group specific and coded by a gene Cry1AC. Among the 3 genes, they have given 1 gene. So this statement is correct. And the next one, the Bt cotton exists as inactive protoxin in Bacillus thuringiensis. However, after ingestion by the insect, the inactive protoxin get converted into active form due to acidic pH of the insect gut. Actually, in the insect gut, we don't find the acidic. We find the basic. In humans, we find the acidic pH in the gut. So, the statement is wrong. So, the correct answer will be statement 1 is true and the statement 2 is false because of this one word acid. So, if instead of acid it is a base, then this statement would be correct. So, the correct answer will be the statement 1 is true and the statement 2 is false. So, we will go to the next question. So, now the next question is from anatomy of flowering plants. So, anatomy of the flowering plants. So, coming to the statement 1, the parenchyma is living. Yes, the parenchyma is the living tissue, but collenchyma is dead tissue. So, here there is a wrong thing. Collenchyma will comes under living mechanical tissue because its corners are filled with cellulose, hemicellulose, pectin. So, this is the wrong thing that has given in the statement. Collenchyma is the living mechanical tissue. So, coming to the next statement 2, gymnosperms will be lacking xylem vessels, right? Gymnosperm will be lacking xylem vessels, but the presence of xylem vessels is the characteristic feature of the angiosperms. Yes, right. The statement is absolutely right. We always differentiate the angiosperms from the remaining plants with certain characteristic features, which among them presence of xylem vessels. So, in this, the statement 1 is correct, wrong, and the statement 2 is correct. So, the statement 1 is false, but the statement 2 is true. So, second option is correct. Moving to the next question. So, this is also the statement based question. This is chromosome. So, this question is from the cell cycle and cell division. The chromosome is visible under the light microscope during leptotein stage. This is the actual phrase given from NCERT. Okay, and the first statement is right. Under light microscope, we can see the chromosomes, which is quite visible. And coming to the statement 2, the beginning of diplotin is recognized by dissolution of synaptonemal complex. We all know the marking point of the diplotin stage will be chiasmata. When the chiasmata will be visible, when there will be a repulsion of the chromosomes. When the chromosomes are getting repelled, moving apart, then we can see very clearly the chiasmata. So, when the chiasmata will become visible, when the dissolution of synaptonemal complex is happening. So, this statement is also right. So, both the statements are right. So statement 1 and statement 2 are true. So option 3. Moving to the next question. So this is the match the following from cell, the unit of life. So coming to the A, nucleolus. The nucleolus, what is the functions? Based on the functions they have given in the list 2. In the list 1, we have the cell organelles. In the list 2, we have the functions of that referred cell organelles. So let's match it. So nucleolus. Nucleolus will be site for active ribosomal RNA synthesis. Assume that if you have more nucleolus, that means more RNA synthesis, that means more protein synthesis. Next, centriole. Centriole is for organization like cartwheel. This is also directly from NCERT. Leucoplast. Leucoplast is meant for storing the nutrients. So for example, we have amyloplast, areoplast, eleoplast. Amyloplast is for storing starch. Areoplast is storing proteins. So like this, leucoplast are the colorless plastids for the storing nutrients. Golgi apparatus. Golgi apparatus, site for the formation of glycolipids. We all know the Golgi apparatus will be having the marker enzymes, which we call it as glycosyl transferases. Actually, the Golgi apparatus is the site for packing of the lipids and packing of the proteins. The lipids are packed as glycolipids. So the right option for this is A, it is 3. So you can see, look for these two options. First initially A is 3 and B is 2. Right, option 3 is correct for this. We'll move on to the next question. List of endangered species was released by. We have options. We all know this is a very easy question. It is IUCN. So I'll expand IUCN. International Union of Conservation of Nature. International Union of Conservation of Nature. So they are the one will be releasing the red data book. In the red data book we have the list of endangered species. So I'll expand once the FOAM and GEAC and WWF also. FOAM, Friends of Arcata Marsh, and GEAC, Genetic Engineering Approval Committee. WWF, Worldwide Fund for Nature. Okay. So these are the options. So but the correct option is IUCN. List of endangered species, which is giving the red data book. So in the red data book, which is given by IUCN, International Union of Conservation of Nature. Move on to the next question. So let's go on to the section B. So we all know how we have to choose the section B as well. The section contains 15 questions. You have to attempt any 10 questions. Student attempts more than 10 questions. If a student attempts more than 10 questions, which he have attempted will be checked. So we all know the rules and regulations for section B. We are moving on to the question. The DNA represented in the chloroplast. We all know this is a common thing for bacteria, chloroplast, mitochondria. In all these three things, the common thing what we find about the genetic material will be double-stranded circular DNA. Okay. So the correct answer for this is circular and double-stranded. Right. So bacteria and chloroplast and mitochondria, we all know chloroplast and mitochondria are the semi-autonomous cell organelles. They have their own genetic material. The very own genetic material will be none other than circular and double-stranded. Move on to the next question. Coming to the next question. Which of the following are fused in somatic hybridization involving two varieties of plants? Excuse me. Protoplast. The right answer is protoplast. For this, the answer will be, usually in case of somatic hybridization, which is done in vitro, means in the laboratory. The two protoplasts, in case of somatic hybridization which is done in vitro, the two protoplasts, other than reproductive cells, okay, other than reproductive cells, they both are fused. The best example for this is potato and tomato. So the both vegetative cells protoplast are fused for somatic hybridization. The correct answer is protoplast 1. Move on to the next question. Identify the correct description about the given figure. So this question shows us how the figures of NCERT is very, very important. Like if you see the answer, we all know this is the corn cob, inflorescence, the racemose inflorescence of the corn cob. If you see this picture, the answer is the description of this picture. So the description of this picture is wind pollinated plant inflorescence showing flower with well exposed stamens. You can check in the NCERT also. Directly below the figure, this is the description that is given. So far this is one of the example. We should not even neglect the pictures of NCERT also. Yes. And why the remaining options are wrong? Cleistogamy. These are not closed flowers, and these are not complete autogamy, and these are not water pollinated. Right. So the very appropriate answer, even in the comes under the description of this picture, is option 3. The wind pollinated plant inflorescence showing flowers with well exposed stamens. Move on to the next question. Yeah, spraying the sugarcane crop with which of this following plant growth regulators increase the length of the stem, thus increasing the yield? So the right answer is gibberellic acid. So it will increase the crop production to 2 tons per acre. So this is also given in NCERT. The correct answer, by increasing the internodal length. Right. Move on to the next question. So this is again the match the following. This time you have many of the match the following. If you find out 1 or 2, you can find out the answer. In that way the questions are quite easy with match the following. So here they have given the list of scientists. This is the question. This is the question actually from molecular basis of inheritance. So Frederick Griffith, he is the one who has given the transforming principle, who worked on Streptococcus pneumonia. And Francis Jacob and Monod, they have given the lac operon concept, gene expression. And next, Hargobind Khorana, he is the one regarding the genetic code. Michelson and Stark, semi-conservative method of DNA replication. Right? So for A it is 3. And for B it is 4. Yeah. If you find any 2, you will be getting the answer very easily this time. So for Frederick Griffith it is transforming. And for Jacob and Monod it is lac operon concept. And Hargobind Khorana is genetic code, and Michelson and Stark it is semi-conservative method of DNA. Next. Again it's a match the following. The GLUT4, which enables the glucose transportation into the cells. So this is the question from biomolecules. And insulin is an hormone. Trypsin, it is an enzyme. Collagen, intercellular material. Intercellular matrix material. So A is 4 and B is 1 and C is 2 So obviously the correct option is option 3. Next, we'll move on to the other question. So again, we got a statement question. In C3 plants, some oxygen binds with rubisco, ribulose bisphosphate, carboxylase and oxygenase. We know already this is the most abundant protein in the entire biosphere. Hence, CO2 fixation is decreased. So this statement is right. Usually the photorespiration, when oxygen combined with rubisco, we call it as photorespiration. Obviously the photorespiration decreases the CO2 fixation. This is exactly the right statement. And coming to the next statement, in C4 plants, the mesophyll cell shows very little photorespiration, while bundle sheath cells shows do not show photorespiration. In the second statement, it is quite tricky. We know the mesophyll cells of C4 plants. You have C4 plants. We have two types of cells. One is mesophyll, another one is bundle sheath. Right? So we all know from NCERT that mesophyll cells will be having pepcase. So if mesophyll cells having pepcase, how the oxygen will be binding with the pepcase? There is no chance for the oxygen for binding with the pepcase, and there is a chance of binding the oxygen in the bundle sheath cells, but it won't allow. So there will be no photorespiration in case of C4 plants. So the statement 2 is wrong. So statement 1 is true, but the statement 2 is false. This is the right statement. This is the right answer. Option 1 is right. Moving on to the next question. 143. Identify the step in TCA cycle which does not involve the oxidation of substrate. If you know the Krupp cycle very well, then this is the question. This is the very easy question. If you see, the correct answer is succinyl-CoA to succinic acid. In this reaction, for the option 1, there will be formation of GTP. Right? There will be the formation of GTP. How the GTP is formed? GDP plus PI. Actually, this phosphate is actually taken from the substrate of succinyl-CoA. So this is the substrate level phosphorylation, and this is not the oxidation. Right? Coming to the other options, the isocitrate to alpha ketoglutaric acid. Here actually in the second option, there will be a release of NADH2. And in the third option, there also will be a release of NADH2. In the fourth option, there is a release of FADH2. All these three is denoting the oxidation, except the succinyl-CoA to succinic acid. This is not the oxidation. This is substrate level phosphorylation. We'll move on to the next question. This is again a match the following. You have very bunch of match the following in 2024 NEET paper. So coming to next, citric acid cycle. So they have given the list of cycles and pathways, and they have asked the site of these cycles and pathways. So coming to the citric acid cycle, it happens in the mitochondrial matrix. So for A it is 2. Obviously if you find out the A, you got almost the answer. And glycolysis. Glycolysis will happen in the cytoplasm, right? And next, electron transport system. ETS will happen in the inner mitochondrial membrane, and proton gradient will happen in the intermembrane space of mitochondria. So for A it is 2 and for B it is 1. So obviously this is the answer, right? Move on to the next question. Which of the following statement is correct regarding the process of replication in E. coli? We all know the genetic material in the E. coli is circular and double stranded. So coming to the statements, so statement 1, the DNA dependent DNA polymerase catalyzes the polymerase reaction in 5 prime to 3 prime as well as 3 prime to 5 prime, whether it is a replication or a transcription. Remember, it won't do in both the direction, it will be doing only in one direction, that is only in the 5 prime to 3 prime direction, that is for sure. So this statement is wrong. And next, the DNA dependent DNA polymerase, the DNA dependent DNA polymerase catalyzes the polymerization in the direction of 3 prime to 5 prime. This statement is also wrong. We already know that catalyzes the reaction only in 5 prime to 3 prime, not in 3 prime to 5 prime. So this statement is also wrong. And next, the DNA dependent DNA polymerase catalyzes the polymerase reaction in one direction, that is 3 prime to 5 prime. No, this is also wrong. So the correct statement is, sorry, one second. The DNA dependent DNA polymerase catalyzes in only one 5 prime to 3 prime. So this is misprinted. 5 prime to 3 prime. Because here you have to check the replication, because replication is that comes to DNA to DNA. Remember, the replication is none other than DNA to DNA. So this is the statement 3. DNA dependent DNA polymerase. This is the enzyme which catalyzes the polymerase reaction in one direction, that is 5 prime to 3 prime. Okay. So option 3 is correct. Moving on to the next question. So coming to this question, this is so far tricky, and in an ecosystem, if the net primary productivity of a first trophic level, we all know that the first trophic level produces, trophic level 1 is producers, trophic level 2 is consumers, this is primary consumer, and trophic level 3 is the secondary consumers. So we all know the gross primary productivity and net primary productivity. So GPP is the gross primary productivity minus respiration is equal to net primary productivity. So in this question they have given the NPP of the primary, so which is the first trophic level producers. The NPP of the producers is 11x kilocalorie per meter square per year. They are asking what would be the GPP, gross primary productivity of the third trophic level of the same ecosystem. That means this level, they are asking this third level, third trophic level. So actually they have given kilocalories per meter square per year. We can follow the energy which is transferred only 10% from one trophic level to another trophic level, right? For example, here for producers, it is 100x, and for primary consumers 10% of this, which is 10x, and for secondary consumers, which is the third trophic level, it could be x. So x kilocalorie per meter square per year. But we have to review this question a bit more. We will be waiting for the NTA final key, as so far we are assuming this would be or this may be the answer, but we are keeping this a hold. This is a little controversial question and this is a bit beyond the NCERT. So we are keeping this question into hold and we'll wait for the final key which is released by NTA. So as so far this we are assuming. Okay, we'll move on to the next question. Coming to the next question, here they have given in the list 1 and list 2 the examples and the types of aestivation or the types of flower. Coming to the rose. Rose is perigynous. We already know plum, peach, rose. These three will comes under the perigynous. The perigynous is neither inferior nor superior. Coming to the next P, marginal placentation. Cotton, twisted aestivation. Mango, with the stony endocarp, this is the drupe. So, obviously the option will be, for A it is 2 and for B it is 4. Yeah, if you find out any 2, you can find out the answer very easily. Okay, so for cotton it is twisted, right? For mango it is drupe. We'll move on to the next question. This is again one more match the following. So, so here they have given the list of scientists and their contributions, right? Robert May. According to the Robert May, the global species that is so far identified is nearly 7 million. And Alexander von Humboldt, who has given the species area relationship. You will be having a rectangular hyperbola graph. And Paul Ehrlich. So Paul Ehrlich has given the rivet popper hypothesis. So, and David Tillman has given the long-term ecosystem experiments. So long-term ecosystem experiments, he is the one who demonstrated the more diversity gives to more productivity. Right? So we'll see the correct option. For A it is 3 and for B it is 1. So obviously option 4 is the correct. Next, question number 149. Here they have given the types of stamens and types of androecium arrangement in the list 1, and in the list 2 you have the examples for that androecium arrangement. So coming to the monoadelphous condition, China rose, which comes under the family Malvaceae. Diadelphous P, which comes under the family Fabaceae. Polyadelphous citrus, epiphyllous condition, which is lily. So for option A it is, for A it is 4, for B it is 2, sorry, for option A it is 4, for B it is 2. Option 3 is correct. And polyadelphous is citrus. And next, okay. Next, coming to the last question of botany. So here we have the statements. We have to choose the correct statements of Phaeophycia, which is the brown algae. So let's see the characteristic features here. What has given from option A to E. Let's find out which one is correct. Asexual reproduction occurs usually by biflagellated zoospores. Yeah, the asexual spores regarding the algae is none other than biflagellated zoospores. This is right. Sexual reproduction is by oogamous method only. No, it has all the three: isogamous, anisogamous and oogamous. Okay, so this statement is wrong. This you can apply to Rhodophycia, red algae, not to this. The stored food is in the form of carbohydrates, which is either mannitol or laminarin. Yeah, the statement is right. The major pigments found are chlorophyll A, C, carotenoids and xanthophylls. Because of this reason that is brown in color. The statement is also right. Vegetative cells have cellulosic wall, usually covered on the outside by gelatinous coating of algin or alginic acid. Right. So all the statements are right except the statement B. So except the B, so A, C, D, E. All these 4 statements are right except the option B, except the statement B. Okay. Yes, right. This is all about the botany paper discussion. So I hope you got many match the following questions and statement questions this time. So the paper is very well, and the paper is entirely strictly from NCERT except the one question we kept on hold. We'll see for the NTA key. Okay. Now the next zoology paper discussion will be starting, and I am handing over to Bhavana ma'am. Okay. Yes.
పెట్టుకున్నాను చేతిలో పట్టుకోండి ఇలా పట్టుకోవాలి మరి ఎలా రాస్తాము నో యూ కెన్ పుట్ ఇట్ హియరా పాకెట్ ఉందా పాకెట్ లేదు లెగిన్ ఉంది స్ప్లిట్ కాదా అవ్వదా ఇది ఎట్లా ఓపెన్ చేయాలి యాక్చువల్లీ కానీ ఇది కష్టం కష్టం జారిపోయి పడిపోతుంది చేతితో పట్టుకోండి ఎట్లా ఏం కాదు మామూలుగా పట్టుకోనా ఇది యాక్చువల్లీ ఈ బటన్ ఇలానే మొత్తం నెక్స్ట్ క్వశ్చన్కి ఇది ఓకే ఇప్పుడు వాయిస్ వస్తుంది వాయిస్ వస్తుంది ఇక్కడ పెట్టుకోకండి ఇక్కడ చాలు మీకు ఇది టైట్గానే ఉంది కదా దగ్గర గట్టిగా ఉంటుంది కిందకి పెట్టుకో పెదాల దగ్గర ఉంది అంతే ఓకే ఇది పట్టుకొని ఉండాలా ఓకే ఫస్ట్ క్వశ్చన్ ఏదొచ్చింది సార్ చూడండి ఒకసారి దిస్ ఓన్లీనా ఫస్ట్ క్వశ్చన్ వచ్చిందా ఏదొచ్చింది ఫస్ట్ క్వశ్చన్ కరెక్ట్ ఆర్డర్లోనే ఉంది కదా ఏంటి ఇట్లా ఇచ్చారు ఓకే ఆప్షన్ లేదు
Hello all, welcome to Zoology Resonance NEET session 2024. This is Dr. Bhavna, Zoology faculty. Today we are going to discuss few important NEET questions. So here, whatever are discussed in the paper. So here, let us move into these questions first. The first question is only the match the following. We are going to discuss the T3 paper and this typhoid. What is typhoid is basically it is the bacterial disease. Typhoid is the bacterial disease. Leishmaniasis, it is the protozoan disease. Next you have the ringworm disease, which is the fungus disease. And next you have the filariasis, which is a nematode disease. So the option is which are the infections which are causing these. So here for typhoid it is the bacterial infection, for leishmaniasis it is the protozoan, ringworm it's the fungus, and filariasis it is the nematode. So what is the option here is, the option is the fourth option. Okay, next let us move into the next question. Next question is based on the human reproductive system. In these about the intrauterine devices they have asked. So in non-medicated IUDs, it is the Lippis loop. Lippis loop is the best example for the non-medicated IUDs. Next you have the copper releasing IUDs. In copper releasing IUDs, there are multiple examples like CUT, CU7 and multi-load 375. But here they specified only one option, which is multi-load 375. Next, the example of hormone releasing IUDs. So in this hormone releasing IUDs, the best example which is given is LNG20. You have another example also which is progesterone, but what here they asked is exclusively it is LNG20. Next, implants. Implants are basically progesterones. So progesterone is the best example here given and it is 2. So the option comes as the second option. Now let us move into the next question. In these, it is a statement type of question. The presence or absence of hymen is not a reliable indicator of virginity. So that is the correct option. It is not a reliable indicator for virginity. Then the statement 2 is hymen is torn during the first coitus only. But here the hymen is torn before only, even before the first coitus, even before the menstrual cycles, that is during cycling and all it will be torn. So that is false. So the statement 1 is correct and the statement 2 is wrong. Now let us move into the next question. So again it is a match the following. For pons, what are the functions here they are asking in this question? So what is the function of pons? It will connect the different regions of the brain. That is the main function of the pons. Next you have the hypothalamus. So what is the hypothalamus? Here they specified about the neurosecretory cells. So it is associated with that of pituitary gland. Hypothalamus basically have the neurosecretory cells in the pituitary gland. Next you have the medulla. What is the function of medulla? All the centers are associated here in the medulla. Like you have respiratory center, gastric center, then you have cardiovascular center. So here they asked about only the respiratory center and gastric center. So next you have the cerebellum. Cerebellum, what is the use is it provides the additional space for the neurons. So what is the function is it provides the additional space. So the option is 3, 4, 2, 1. 3, 4, 2, 1 is the fourth option. So let's move into the next question. Which of the following is not a steroid hormone? See, all are steroid hormones only. Like we have the progesterone, cortisol, then testosterone, everything is a steroid hormone. Only one which is a peptide hormone is glucagon. So here they are asking which of the following is not a steroid hormone. So the answer is glucagon. Okay, let's move into the next one. Okay. So okay. There is a small thing after this question. You have the end here. ఆ స్లైడ్స్ అక్కడ వరకే ఉన్నాయి. Only 6 questions slides were given. I don't know. That live continued. You paused there. See, this is ending. That is only the problem. Only 5 questions they have. ఇంకేమైనా చేంజెస్ చేయాలా మ్యామ్? ఇందిరా మ్యామ్, ఎనీ చేంజెస్ టు బి డన్? No, in mine any changes we have to. I'm turning that side or this side. First suddenly it's like that is not working properly. Huh? Here, where is my phone? You kept inside. Once get now, ma'am. Slides like ఎక్కడ ఆగిపోయింది ఎగ్జాక్ట్గా లైవ్ నేను చేస్తున్నప్పుడు ఆగిపోయిందా 156 క్వశ్చన్ కానీ నేను చెప్తున్నా ఏంటది ఆ మధ్యలో స్లైడ్స్ లేవు స్లైడ్స్ ఆగిపోయాయి సార్ ఇప్పుడు చెక్ చేసి పెడదాను సార్ మళ్ళీ బాగోదు నా suddenly I was thinking it was my mistake. How many times? How many questions are there? 6 only I discussed. Now here in this presentation. Daddy. Okay. 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 70 after that? Yeah, where is the present? They are getting I think. Okay, let us move into the next question. Which one is the correct product of DNA dependent RNA polymerase? So here what they have given you is from 3' primer. See, they have given from the next question 3' prime to 5'. So here everything is the template strand which we are discussing. So this is the DNA dependent strand and next they are asking about the RNA dependent. So obviously it will start from that of 5' to 3', and here when it starts from 5' to 3', the option will be as for T it is A, then for A it is U. Because it is RNA, so for adenine it is uracil which is present. Like that if you see the option, then the option will be as 3. Third option will be correct. Because for T it will be A, for A it will be U. So that is the next question. Let us move into. Here there are 3 types of muscles given as A, B and C. Identify the correct statement along with their location in the human body. So here you have skeletal muscle, smooth muscle and cardiac muscle. By name only here we can understand by seeing this picture. So this is the skeletal muscle, this is the smooth muscle and this is the cardiac muscle. So where is skeletal muscle present is important. See here in the first option you have the biceps. That is correct. Next in the last option you have the triceps. It is correct. But the second is smooth muscle is involuntary. Everything is correct. But in the third option what do you have is heart. Heart is basically cardiac. So the option goes as the fourth option. Okay, let us move into the next one. Following are the stages of the cell division. What are the stages of the cell division you have to see. See some questions, even botany questions are asked in zoology paper like these questions. But we have to answer as we read both the botany and zoology. It is combined biology you get. So here you have the G1 phase, next S phase, G2 phase and the last karyokinesis. Karyokinesis and finally you have the cytokinesis. So here in these options what is the sequence they are asking. So we have to tell in karyokinesis only all will be included like prophase, metaphase, anaphase and telophase. Everything will be included in this. So the option first you have to see. So the option first is G1 phase, that is E option. Only in these two you have. Then you have C, that is synthesis phase. So this goes as the option. G1 S phase, G2 phase, karyokinesis and it is cytokinesis. Okay, let us move into the next question. Which of the following are autoimmune disorders? Autoimmune disorders means nothing but which attacks our self cells. So in these autoimmune disorders, myasthenia gravis is the one disease which will affect our own cells. Then you have rheumatoid arthritis which affects, but gout is not the autoimmune disease. Of course it is the inflammation of uric acid crystals, but it is not an autoimmune disease which you get. Muscular dystrophy is also not an autoimmune disease. Then what is muscular dystrophy? It is the genetic disorder which is affecting the muscles. Next you have SLE. SLE is nothing but systemic lupus erythematosus, where you have multiple swellings in the body, and this is an autoimmune disorder. So which of the following means A, B and E? So the option is fourth one. Okay, let's move into the next question. Again this is the match the following question. So here in lipase, which bonds are present, they are asking. This is a biomolecules question, and this lipase means nothing but lipids. In lipids, which type of bond you will have is ester bond. So this option is 2. Next you have nuclease. In nuclease it is nothing but the nucleic acids. So what are the bonds associated here is phosphodiester bond. Next you have protease, which is associated with proteins. So here directly you can write it as peptide bond, and then cerebellum. So here I think the option is given as wrong. So lipase, nuclease and protease, first we have to see. So 2, 4, 1. 2, 4, 1, 3 is the option here. Okay. So let's move into the next question. The flippers of penguins and dolphins are the example of. Flippers of penguins and dolphins are example of convergent evolution. It is nothing but they are analogous organs. What is the meaning of analogous organs? That is, they have same function but different structure. So which evolution type they belong to is important. So they belong to that of convergent evolution. If they would have asked, suppose sweet potato and potato also, it is analogous organs only. So here we are seeing about the convergent evolution. Okay. Next, this is an match the following of breathing and exchange of gases. Here they have asked about the expiratory capacity. What is expiratory capacity? First you have to understand. That is tidal volume plus ERV. That is expiratory reserve volumes give you expiratory capacity. So the option is 2 here. Next you have functional residual capacity. In functional residual capacity, it is ERV plus RV is the functional residual capacity. Vital capacity, all the three you will be having, like TV, IRV and ERV. All the three you will have here. So vital capacity, the option here is 1. Inspiratory capacity. So for inspiratory capacity it is TV plus IRV. So here based on this option we have to see it in right. So the option first here is you have 2. Then functional residual capacity, that is you have the first option is wrong. So 2, 4, 1, 3 is the correct option. This is associated exclusively of volumes and capacities associated with the lungs. Okay. Then next you have which of the following factors will not affect the Hardy-Weinberg's equilibrium. Which will not affect the Hardy-Weinberg equilibrium basically is constant gene pool. Because all other factors, like we have gene migration, where the migration will take place of the habitat. Next you have the genetic recombination, where the frequency of alleles is changing. Then you have the genetic drift. That is one population moving to another. So these all are the factors which will affect the Hardy-Weinberg's equilibrium, and which will not affect is constant gene pool which you have. So again the question of human evolution only they have asked. So here they are asking the evolution of man stages. What are the stages associated with human evolution of man? So initially you have Homo habilis, then erectus, Next, Neanderthal and Homo sapiens. See, there is an order given directly from the NCERT. This order is given like you have how the species have evolved. So here the option is Homo habilis, then you have Erectus, then you have Neanderthal, finally we are called as Homo sapiens. Okay, so the option is second option. Okay, let's move into the next question. Following are the stages of pathway for conduction of action potential throughout the heart. How the action potential is conducted? See, as we know, see here, when we draw the heart, first from the sinoatrial node is the conducting system. Next we move into the AV node. Then you have AV bundle here. Yes, then you have the division, that is the bundle of his. And finally here you have Purkinje fibers. So they are asking how the order or the impulse is transferred basically. So here first initially this is the first one. So the option is E, sinoatrial node. Then you have atrioventricular node, that is C. Then you have AV bundle as I told you. Next you have bundle of his, and finally it is the Purkinje fibers. So the option is third option for you. Which of the following factors are favorable for the formation of oxyhemoglobin in the alveoli? So what are the factors which favor the combination of oxygen for hemoglobin is exactly you have to say that high oxygen content should be there. High oxygen should be there and high pH should be there. What are the options here? Everywhere you have to see where is high oxygen levels. So high oxygen levels is there, and here two options are given like this. But here high PCO2 and lesser H+. So lesser H+ is correct because I told you only two, that is oxygen levels and pH levels should be more. But here what is happening is it is not the pH level which is more, then other factors all will be less. So when other factors are less, it is lesser H+ concentration. Okay. So the option is fourth option. Next again you have the match the following. Today basically more of match the following questions are given in your zoology part, and here the first one comes as in biotechnology also the match the following. So alpha-1 antitrypsin. So alpha-1 antitrypsin basically it is, what is the one which is related to is, it is related to emphysema. Next you have cry1AB. That is cry1AB is nothing but it is cry gene, all of you know, right? What is the cry gene? It is associated with the beta toxins. So beta toxins plant Bacillus thuringiensis. So here which is for what they are asking, that is for cry1AB is exclusively only for corn borer. Then cry1AC is associated with cotton bollworm, and then you have enzyme replacement therapy. Enzyme replacement therapy means nothing but it is associated with ADA deficiency. In few children like ADA deficient enzyme will be there. For them enzyme replacement is needed, otherwise they will die. So here what are the enzymes which are associated is important. So the option goes as 3412. So the option is first option 3412. Now let us move into the next question. Again this is an assertion and reason type of question. In assertion, FSH act upon ovarian follicles in female and Leydig cells in male, right? What is FSH hormone? First you have to understand. It is follicle stimulating hormone. So what is this follicle stimulating hormone? It will basically regulate, in males it will act, in females also it will act. Suppose if it act in males exclusively, then it is it plays an important role in ovarian follicles. Suppose if it act in males, then these release one hormone which is androgens. So FSH act upon ovarian follicles and Leydig cells in male, that is correct. Then reason is growing ovarian follicles secrete estrogen in female, that is correct, while interstitial cells secrete androgen in male humans, that is also correct. See, both are correct, but it is not the correct explanation, because FSH we are talking about differently and growing ovarian follicles we are talking different. So it is A and R are true and R is not the correct explanation of the A. Now the next question. The following diagram showing the restriction sites in E. coli cloning vector, that is PBR 322. So we know that in PBR 322 this is the structure. Already they have given the structure of PBR 322. Only what they are asking is X and Y. This is actually, this X and Y is basically constant. X is nothing but the ORI site and Y is nothing but the ROP site. See, ORI site means we have to understand that it is associated with origin of replication. Then ROP site is associated with that of proteins. So here the answer is X is responsible for controlling the copy number of the DNA and Y is responsible for replication of the plasmid, that is the protein which is associated. So here are these questions which we discussed, and you know that it is basically zoology direct NCERT questions are given. Of course few botany questions are mixed up, but it can be answered. They all are the easy questions. Okay, the next set of questions will be taken by our next faculty, Mahima ma'am. Okay, thank you.
What is this not? Choose is not here.
It's not there. They didn't give. They did not type actually.
It's wrong.
Only
Check
answers
the
are
next
there.
slide.
Column is not there.
Sir,
It is
check the
missed.
next slide. But the previous one is wrong, sir. It's complete question ralelo. Shall I start from 172 only?
Yeah, start off. Otherwise, again they have to print and come. It is difficult.
What next? Can you just show me? 172 from 172 show me. Yeah, okay, next. 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 Done. Fine. Okay, done. Yeah, just hold it in my hand. Sir, shall I start? Yeah. Hello students, so now we'll discuss the next set of questions. So here you can see, consider the following statements. Annelids are true coelomates. Okay. Now the porphyrins are pseudo-coelomates. Are they? No, they are coelomates here. Okay. Yeah. Next, the Astracohelminthes, they are coelomates. No, they are pseudocoelomates. Now this is also wrong. Okay, so the Platyhelminth is a pseudocoelomate here. Okay, so it's a coelomate again. So even this is wrong. So here they are asking which option is correct. Okay, so our answer would be the fourth, only the first one. Okay, next. Now given below are the two statements. The statement 1 says in the nephron the descending limb of loop of Henle is impermeable to water and permeable to electrolytes. It's wrong. It is permeable to water but it is impermeable to the electrolytes. Coming to the second statement, the proximal convoluted tubule is lined by the simple columnar. No, it's cuboidal. Brush border epithelium, and it decreases surface area for reabsorption. So here choose the correct answer for the given. So the statement 1 is true but the statement 2 is false. No, as we can see both here are false, so we can directly mark both the statement 1 and 2 are false here. Next. Yeah, so the next question, the joints here. Okay, so the fibrous joints, we know the fibrous joints don't allow any movement, so directly you can mark it. It's the third one. The cartilaginous joints here, so here in the options you can see the third is here. Okay, next the cartilaginous joints, you can see it's like allows the limited movement. Okay, so it would be the first one. The hinge joints, they help in the locomotion. You observe it mainly in the knee. And the ball and socket joints, it's in the humerus, okay, and the pectoral girdle, which allows the rotational movement. Okay, next. So which of the following is not a natural or traditional contraceptive method? So here the lactational ammonia is a natural, and the ones it's not a natural. Okay, the coitus interrupts and the periodic abstinence is also not a natural or the traditional contraceptive methods. Coming to the next question. So the match the following, you have the direct question even here. So the Purobractis, you observe it in the Pterophora. Okay, so the radula, you observe it in the mollusk. Okay. And next the stoma cord, you observe it in the Hemichordata. Okay, and the air bladder, it's present in the Ostrictis. Now here the option A is 2. Okay, and the B is 1, the C is 4, and the D would be 3. So we can go with the fourth option. Next question. So which of the following statement is incorrect? So here the bioreactors are used to produce the small scale bacterial cultures, which is not true. Okay, so here it's not scale, like it's a large scale, the capacity is from 100 to 1000. So next, the bioreactors have an aggregator system, an oxygen delivery system, and foam control system, which is true. Next, a bioreactor provides optimal growth and growth conditions for achieving the desired products, which is also the true. And the most commonly used bioreactor of stirring type, this is also true. But here they are asking which is the incorrect one. So we can go with the first option. Next. Okay. Yeah. So here we can see again a direct question. So the diakinesis where we can observe. Okay. So here in the so here we can observe it in the completion. The chromosome look like thin threads. Next, the pachytene, you can observe the synaptimal complex. The zygotine here, the complete terminalization of the chiasmata, and the liptotine is the fourth one. You can go with the option 1. Next, breastfeeding. Now here assistant and reason 1. So in the first session, the breastfeeding during initial period of infant growing is recommended by doctors for bringing a healthy baby, which is true. Next, the colostrum, they contain several antibodies which are absolutely essential for developing resistance for the newborn baby. Okay, so both are right. So and even it's a correct explanation, so we can go with the third one. So both A and B are correct, and DR is the correct explanation of A. Okay. Now here the TI plasmid which we see in the agrobacterium. So what the TI stands for? So here the TI stands for the tumor inducing. It doesn't stand for the temperature independent. It doesn't stand for tumor inhibiting. It doesn't stand for the tumor independent plasmid. So the answer would be the first one, tumor inducing.
Now, which of the following is not a component of fallopian tube? We know we have three, which is infundibulum, okay, ampulla and isthmus. So obviously the uterine fundus would not be the answer. Next, we have the, again the match the following. So here the Down syndrome, we observe it in the 21st chromosome, okay, and the alpha thalassemia, we observe it in the 16th chromosome. The beta thalassemia, we observe it in the 11th chromosome, and the Klinefelter syndrome, it's in the the X chromosome. So we can go with the option 1. So the next set of questions would be discussed by Indra ma'am.
It's okay. First, you messed up. Search cheyna. Running lone unda. Okay. So hello all. So we have already come to the end of this biology discussion, biology analysis. So last 15 questions I'm going to discuss. So let's look at this 186th question. So the question is saying, which of the following is not a component of fallopian tube? See, if you look at the fallopian tubes, so this 184 question we have already discussed, okay, 186th. So the following are the statements about non-chordates, okay. So what are the features of chordates and what are the features of non-chordates? So if we recall, if we try to recall the characteristic features of the chordates, so the 4 to 5 characteristic features are there. The very first one is the having the postanal tail and the dorsal central nervous system, dorsal spinal cord, the dorsal hollow nerve cord will be there, and notochord will be present, pharyngeal gill slits are also present, okay. So let's look at these options. First one is pharynx is perforated by gill slits, that is nothing but they are referring to the pharyngeal gill slits, okay. So the first option, pharyngeal gill slits, so this is the feature of chordates. This is a feature of chordate, but the question is asking what are the statements, the following are the statements about non-chordates. They are asking about the characteristic features of non-chordates, not the chordates, okay. So the first option is regarding the chordate it is one of the characteristic features of the chordates. So this is not the option. Now look at the second option, that is the notochord is absent. But if you observe the chordate features, what are the characteristic features of the chordate? The chordates will have the notochord. So the second statement is notochord is absent. That means it is not a chordate character. So it is a characteristic feature of a non-chordate. Okay, so this could be the option. This is the option. Next, central nervous system is dorsal. What do you mean by central nervous system? What exactly is present in the central nervous system? Your brain and then the spinal cord. Okay. So now if you observe in our body, in the mammals, in us, okay, in the human beings, the spinal cord will be dorsal. It is placed dorsally or ventrally? It is dorsal, okay. So the central nervous system is dorsal in case of chordates. We human beings are the chordates, okay. So this is a chordate feature. This is a chordate feature. And heart is dorsal if present. If you look at if you observe in our body, the heart, the position of the heart in the human beings also, in human beings are nothing but they are the chordates, okay, we are the chordates. And in our body, the position of the heart is ventral, not the dorsal. So chordates will be having the ventral heart and non-chordates will be having the dorsal heart. So this is not a characteristic feature of chordates. This is a characteristic feature of a non-chordate. Now coming to the last option, that is post-anal tail, is absent? I've told you in the beginning itself that post-anal tail is one of the features of this chordate characters, okay, the chordates. So if this post-anal tail is absent, absolutely it is not a characteristic feature of a chordate, it is non-chordate. So how many options we have? How many options we have for the as the characteristic features of a non-chordates? 3 options. First is notochord is absent. Notochord is absent in the non-chordates, and heart is dorsal if it is present. Dorsal heart will be seen in case of non-chordates, and post-anal tail is also absent in the non-chordates. So how many options we have? 3 options. What are those? B, D and E. Okay, B, D and E. So this, the first option is your answer. Next, coming to the next question. It is a match the following question. Okay, so list 1 you have the eras, okay, all the eras you have, and in the list 2 you have the what are the organisms originated in that era. Okay, the organisms belonging to those eras, so those organisms have been listed in the list 2. Okay, so let us observe. First one is Mesozoic era. So in the Mesozoic era, what are the organisms originated? Those organisms are nothing but mammals. Okay, so in the Mesozoic era, the reptiles. Pardon? So in the Mesozoic era, the reptiles are nothing but the dinosaurs. Dinosaurs are nothing but they are the reptiles. So in the Mesozoic era, the reptiles are originated, and in the Proterozoic era, so A is 3. In the Mesozoic era, the birds and reptiles are originated. So A could be the third option. A is 3. So in 2 options you have A is 3. Okay, so let us follow the elimination method. Okay, so let us look at the Proterozoic era and Cenozoic era. So Cenozoic era is very easy. Cenozoic era, in what happened in Cenozoic era? The mammals got originated. So Cenozoic era relates to the mammals. So C4. So C4. In these 2 options we have the common option, that is A. If you follow the elimination method, if you look at this Cenozoic era, the mammals got originated, and C4, in which option C4 is present? In this option. So your answer could be 2. Okay, so for this question the answer could be the second option. So in the Mesozoic era, the birds and reptiles got originated. In the Proterozoic era, the lower invertebrates originated. In the Cenozoic era, it's very easy, it's very famous. Okay, so the Cenozoic era is the era for the origination of this mammals. Okay, the mammals got originated in this era. Now look at this Paleozoic era. So Paleozoic era is for the fish and then the amphibian. Okay, the Carboniferous period, the Devonian period, that is the golden age of fishes. Okay, so all these periods are included under this eras. Okay, so this is the answer. The second option is the answer for this question. Now coming to the 188 question, statement 1. So this is based on this is statement based question. So what the statement 1 is saying? The statement 1 is saying the cerebral hemispheres are connected by nerve tract known as corpus callosum. So if what is the largest part of the brain? That is cerebrum. Cerebrum has the right portion and then the left portion. 2 halves will be there for the cerebrum. So right portion and then the left portion, these 2 portions, these 2 halves, we call them as cerebral hemispheres. Okay, so right hemisphere and then the left hemisphere. So both these hemispheres are connected together or joined together with the help of one structure. That structure is nothing but corpus callosum. Okay, so according to this, the statement 1 is true. The cerebral hemispheres are connected by nerve tract known as corpus callosum. So this is true. Now coming to the statement 2. The brain stem consists of medulla oblongata, pons and then the cerebrum. So let me draw the diagram of this brain first. So if you look at the diagram, so you can easily say what exactly is forming the brain stem. Okay, so let's say this is a rough diagram of your brain. Now if you observe, so this is nothing but here you will be having the corpus callosum and then the pituitary gland. The pituitary gland will be there and you have a midbrain over here. Okay, so this midbrain along with the along with this medulla oblongata. This is medulla pons, and this is pons, and this is the medulla oblongata. This is pons and this is the medulla oblongata. So your midbrain and then the hindbrain, midbrain as well as hindbrain, both will be forming the brain stem, except, okay, midbrain plus hindbrain, okay, midbrain plus hindbrain, except minus your cerebellum. Because what are the parts of your hindbrain? Hindbrain is formed by cerebellum, pons, and then the medulla oblongata. So pons, medulla oblongata, and this is nothing but your cerebellum. So all these three together makes the brain. These three together makes the hindbrain. These are the three parts of your hindbrain. So brain stem is formed by the midbrain as well as hindbrain, but you have to exclude the cerebellum. If you look at from here to here, okay, if you look from here to here, if you observe, so this is like a stem of a tree, okay, stem of a tree. You know the stem of a tree, right? So if you look at from here to here, it gives an appearance of a stem of a tree. Okay, so brain stem, if you include the cerebellum, no, you cannot say it is looking like a stem of a tree. Okay, only if you exclude the cerebellum, then only it is appearing as a stem of a tree. Okay, so midbrain and then the hindbrain except the cerebellum forms the brain stem. So what are the parts included under this? Your pons, medulla oblongata, and then the midbrain. Okay, so statement 2 is saying the brain stem consists of the medulla oblongata, okay, true, pons, yes, and cerebrum they are saying, but cerebrum is not at all a part of your brain stem. So what should be included here? Your midbrain. Okay, midbrain will be along with the medulla oblongata, and then the pons except the cerebellum will be forming your brainstem. Okay, so what should be the option for this? So statement 1 is true because both the cerebral hemispheres are connected by the corpus callosum, but statement 2, what it is saying, the brainstem consists of medulla, pons, and then the cerebrum, which is wrong, which is false. Okay, so statement 1 is true, statement 2 is false. So where is the option? Statement 1 is correct, but statement 2 is incorrect, the first option. Okay, next, identify the correct option A, B, C, D with respect to spermatogenesis. So they have given a flow chart, and you have to fill what will be in place of A, B, C, D, what will be coming. Okay, so GnRH, what do you mean by GnRH? Gonadotropin releasing hormone. Where it is present? In the hypothalamus. Okay, so hypothalamus will be having all the releasing as well as inhibiting hormones that will be inducing the releasing of hormones or inhibiting the release of hormones from the pituitary gland. Okay, from the pituitary gland. So what is this hormone? GnRH, which is gonadotropin releasing hormone. So it will be inducing the pituitary to release the gonadotropin hormones. What are the gonadotropin hormones? The LH as well as FSH. Okay, so gonadotropin releasing hormone will be inducing the release of this LH. Okay, LH is nothing but luteinizing hormone, and FSH, LH along with the FSH. So two types of gonadotropins. Two gonadotropins will be there. One is LH and another one is FSH. So LH they have already given, but you have to fill in place of A you have to write that is FSH. Okay, which is follicle stimulating hormone. Next, LH, what is its action? What is its function? It will be acting on the Leydig cells. Where are these Leydig cells? Leydig cells are present in the seminiferous tubules. Okay, this is a part of male reproductive system. So luteinizing hormone will be acting upon this Leydig cells. So it will be acting upon the Leydig cells, and FSH will be acting upon the Sertoli cells. If you look at the seminiferous tubules will be having two types of cells. Sertoli cells will be having also the Leydig cells. Okay, Leydig cells are nothing but they are the interstitial cells, and from these Leydig cells the androgens will be released. Androgens are nothing but testosterone. Okay, and from the Sertoli cells, so Sertoli cells are nothing but they will be providing nourishment. They have the function of providing nourishment. So FSH will be acting upon the Sertoli cells, LH will be acting upon the Leydig cells, and Leydig cells will be producing the androgens like testosterone, and formation of spermatids. So everything will be everything will be useful for the formation of the sperms, okay, spermatogenesis. So Sertoli cells will be releasing some factors which are responsible for the spermiogenesis. So what is FSH? So FSH is present in two options, first as well as third. So what should be the what is B? B is nothing but it is a Leydig cell. So if you observe, first as well as third option is having FSH, but in place of B, Leydig cell is the correct option. So in these two options you have Leydig cells in the third option, so your answer could be 3. Okay, so next question, 190. So again there is a match the following question. So list 1 as well as list 2 is given. First, RNA polymerase 3. So if you look at this RNA polymerase 3, so fourth option directly you can say fourth option is the related option, okay, and you have A as fourth, you have only in one option A4 is given, so directly you can follow the elimination method and you can tick this answer 2. Okay, so option 2 will be the answer for this question, and Tata box is for it will act as a promoter. Okay, so this is a very easy question. And next one, again one more match the following question you have. So this is all related to the disorders, okay, disorders in the chemical control and coordination you will be coming across it. So first disorder is exophthalmic goiter, exophthalmic, exo means outside, ophthalmic means it is related to the eyes, okay. So that means in this particular disorder the eyes will be protruded outward. So where do you see that word? Hypersecretion of thyroid hormone and protruding eyeballs, okay. So protruding eyeballs is the key word for this exophthalmic goiter. So exophthalmic goiter, it is due to the hypersecretion. Hypersecretion means excess secretion of your thyroid hormone, and what will happen? The eyeballs will be protruded outward. Next one is acromegaly. Acromegaly, this is due to the hypersecretion of this growth hormone or the excess secretion of this growth hormone. So B will be fourth. So where do you see A as 3? So in two options you have A3 is in two options A3 is given, and if you follow the elimination method again, so for B also we have the common option, but for C we have the different options. So C, in first option C is given 2. What is C? Cushing syndrome. Cushing syndrome is the keyword for this Cushing syndrome is moon phase, okay. So C could be 1. So where the 1 is given? In this option. So in the second option C is given as 1. So this could be your answer. And D is cretinism. Cretinism is observed in the pregnancy, during the pregnancy, and during pregnancy due to the hyposecretion, less secretion of this thyroid hormone causes stunted growth, which is nothing but your cretinism, okay. So what is the option here? Second one will be the answer for this question. Next, again the match the following question. So unicellular glandular epithelium. What are the unicellular glands we have in our body? Only one, that is goblet cell. What is the function of this goblet cell? Where it is present? It is present in the intestine, and goblet cell, the function of this goblet cell is to produce the mucus, okay. So goblet cells are the unicellular glands. So where the goblet cells are given? Third. So A could be third. So A is 3. So only in one option you have A as 3. So directly you can choose the option 1 as your answer, okay. Next given the multicellular compound epithelium. Compound epithelium or stratified epithelium, both are similar. If you observe the stratified non-keratinized epithelium is seen in case of moist surfaces of your body, like moist surface of your buccal cavity. So compound epithelium is moist surface of buccal cavity. Multicellular epithelium, glandular epithelium. So multicellular glands, what are the multicellular glands we have in our body? Salivary glands, sweat glands. Sweat glands as well as salivary glands, both are the multicellular glands in our body. So salivary glands, so they have already given the salivary gland. So this is a this is one example of a multicellular gland. And endocrine glandular epithelium. Endocrine, whenever you see this word endocrine, blindly you can say this is the function of this gland is to secrete the hormones. Okay, so which out of these 4 options, which one will be having the role in the secretion of the hormones? That is pancreas. Okay, so endocrine glandular epithelium is nothing but your pancreas. So what is the option? The first option, of course, this is directly you can follow the elimination method and you can choose the option 1 as your answer, because A3 is given in only one option. Okay, only for your reference I have explained everything. Next, 193. Given below are 2 statements. Again a statement based question. So what statement 1 is saying? Bone marrow is the main lymphoid organ where all blood cells including lymphocytes are produced. Without any doubt, all the blood cells, okay, lymphocytes are nothing but they are the kind of WBC are nothing but white blood cells, so they are also blood cells. So all the WBCs, all the blood cells in our body are formed without any doubt in the bone marrow. Which bone marrow? Red bone marrow. Okay, so bone marrow is the main lymphoid organ. It is a primary lymphoid organ. We have 2 types of lymphoid organs in our body: primary lymphoid organ and then the secondary lymphoid organ. Primary lymphoid organs are nothing but bone marrow and thymus. Secondary lymphoid organs, we have many secondary lymphoid organs like spleen, Peyer's patches, appendix is also a secondary lymphoid organ. Okay, so bone marrow is the main lymphoid organ in which all the blood cells including lymphocytes are produced. This is true. The statement 1 is true. Next, the statement 2. Both bone marrow and thymus provide microenvironments for the development and maturation of the T lymphocytes. Remember this, like bone marrow is the site for the development, for the formation, development and maturation of the B lymphocytes. Okay, but T lymphocytes, they will be, of course they will be formed within the bone marrow because all the blood cells will be formed within the bone marrow. So the same way the T lymphocytes will also be formed within the bone marrow, but for their maturation they have to migrate to the thymus. Okay, so thymus will be the both bone marrow and thymus provide microenvironments for the development and maturation of T lymphocytes. So this is also true. Both the statements are true. So what is the option? Statement 1 is correct. Okay, statement 1, both statement 1 and statement 2 are correct. So this will be your answer. So option 3 is the answer for 193 question. Next, this is also one more match the following question has been given. So list 1, the structures used for, so this is all about the cockroach. Okay, this question is completely related to the cockroach digestive system. Okay, so the structures used for storing of food. So what is the organ, if you observe if you try to recall the digestive system of the cockroach, what is the organ responsible for the storing of the food? That is crop. Okay, so where the crop is given in 4th option. So the structure used for storing food is nothing but crop. Ring of 6 to 8 blind tubules at junction of foregut and midgut. So between the junction between the foregut and midgut, you have 6 to 8 blind tubules. They are nothing but hepatic CK or the gastric CK. Okay, so B2, A4 and A4 and then the B2. In which option you have A4 and B2? So this will be your answer. And let's check the other options also. Ring of 100 to 150 yellow colored thin filaments at the junction of midgut and ingut. They are talking about the Malpighian tubules. So C will be 3. And D, the structures used for grinding the food. That is G for grinding, G for gizzard. Okay, so gizzard is the organ in which the food will be ground and crop is the organ in which the food will be stored. This is in case of the cockroach digestive system. Okay, next. Choose the correct statement given below regarding juxtamedullary nephrons. Okay, so this is regarding the excretory system of your body. So first option, loop of Henle of juxtamedullary nephrons runs deep into the medulla. So in our body, two types of nephrons are there. One is cortical nephrons. I mean, not in our body, in generally 2 types of nephrons will be there. One is cortical nephron, another one is juxtamedullary nephrons. Okay, cortical nephrons are the ones in which the loop of Henle is very short and everything entirely it will be present, mostly it will be present within the cortex region, cortical region. But if you observe the juxtamedullary nephrons, the second type of nephrons. In this, the loop of Henle will be very long and it will be penetrating very deep into the medullary region. Okay. So what is the first statement they are saying? The loop of Henle of juxtamedullary nephrons runs deep into the medulla. This is true. Okay. Next, the second option is juxtamedullary nephrons outnumber the cortical nephrons. No, this is false, because the cortical nephrons are more in number and juxtamedullary nephrons are very less. So juxtamedullary nephrons outnumber means they are more than the cortical nephrons, but this is false, because cortical nephrons are more in number than the juxtamedullary nephrons. Next, the third option is juxtamedullary nephrons are located in the columns of Botany. This is totally deviating option, okay, because columns of Botany is Botany or renal columns. They are otherwise known as renal columns. This is the space between the two pyramids, the renal pyramids, okay. So juxtamedullary nephrons, they are situated more deep into the medulla, not in the columns of Botany, okay. Fourth option is renal corpuscle of juxtamedullary nephrons lies in the outer portion of the renal medulla. This is also false, because renal corpuscle is otherwise known as Malpighian corpuscle or Malpighian body. This is formed by the Bowman's capsule as well as your glomerulus. The glomerulus as well as Bowman's capsule together we call them as renal corpuscle, and this is completely situated within the cortical region, not in the medullary region. Okay. So what is the option? What is the correct answer for this question? First option, which says loop of Henle of juxtamedullary nephrons runs deep into the medulla. Okay. Next, this is again one more match the following question. First one, so this is about ECG, electrocardiography. So here they have given the waves, different waves, and you have to choose what exactly happens what exactly causing this peak to observe in the ECG. Okay, so P wave is observed in the P. So if we draw the diagram, so this will be the P wave, and there will be isoelectric line. So here the negative, 0 and positive. So there will be a isoelectric line. This is the P wave, and you will be having a negative deflection and then the positive deflection, again the negative deflection. So this is nothing but QRS complex, after which you have another isoelectric line and then you have a T wave. Okay. So with this one cardiac cycle will be done. Okay. So P wave, generally it is observed due to the atrial systole or atrial contraction. So P wave or atrial contraction systole, what are the another name of this contraction? Systole or depolarization. So P wave is due to the depolarization of the atria, and QRS complex, this QRS complex is due to the depolarization of the ventricles. Okay, depolarization of ventricles. So depolarization of ventricles. T wave is due to the repolarization or the relaxation of the repolarization, diastole or relaxation, all are similar. So T wave is observed during the repolarization of the ventricles. And TP gap, what do you mean by TP gap? So with this one cardiac cycle is done, and after which again you have a P wave. So between the T wave and then the P wave there will be a gap. This gap we call it as joint diastole, in which no electrical activity will be there. Okay, zero electrical activity will be there. So that phase we call it as joint diastole. So TP gap indicates the heart muscles are electrically silent, because there is no electrical activity, zero electrical activity will be there in the joint diastole. Okay. So what should be the option? P wave A3. A3 is given in only one option, that is fourth. Okay. So fourth will be your answer. Next. This is genetics. Okay, this question is from genetics. So as per ABO blood grouping system, the blood group of father is B positive and mother is A positive and child is O positive. Their respective genotype can be. So what are the options given here? So if you observe the option C, IAIB, which is not a genotype for the B positive. Respectively they are asking. So B positive cannot be, for B positive IAIB cannot be the genotype, because this is the genotype for the blood group AB. Okay, so you can eliminate this, and you can also eliminate the I, IB, IA. Okay, you can also eliminate this E option also, and you can also eliminate the B option, because remember, for the child to have the O positive, O blood group, both the parents should be heterozygous, not homozygous. But if you observe this option IB, IB, IA, IA, II, that is for the O group. Okay, so all are homozygous condition, but homozygous condition can never lead to the O blood group. Okay, because for the child to have the O blood group, the parent should be heterozygous. Okay, there is no other option. So where do you see this heterozygous condition? In the first option, IBI, IAI, and then you will get, when we cross these two genotypes, you will be getting the O blood group. Okay, so first the A option will be the answer for this question. Next, so again the statement based question. So Gauss competitive exclusive principle, this is from the Ecology. So Gauss competitive exclusive principle states that two closely related closely related species competing for different resources indefinitely. No, this is not different, for the same resource. Okay, when the resource is same, only then the competition will be occurring between the two different species or the two similar species. So this is what the Gauss principle states, but here they have given the different resources they have given, but for the same resource the competition will be happening. Okay, so this is false. So the statement 1 is false. Now coming to the statement 2. According to Gauss principle, during competition the inferior will be eliminated. This may be true if resources are limiting. Only when the resources are limiting. Okay, Gauss principle will be applicable, will be obeyed, will be followed only when the resources are limited. And in this competition, who will be winning? Obviously the weaker one will be eliminated and the stronger one will be winning the competition. Okay, so according to Gauss principle, during competition the inferior will be eliminated. That's true, and this may be true if resources are limiting. Only when the resources are limiting, then only the competition will be happening. Okay, so this is true. So statement 2 is true, but statement 1 is wrong. So in which option you have that? Statement 1 is true, but no, statement 1 is false and statement 2 is true. So second option will be the answer. So this is from the biomolecules, so formation of enzyme substrate complex and everything. Okay, first option. So regarding catalytic cycle of an enzyme action, select the correct sequential steps. So we have to choose the correct sequential order of the enzyme substrate complex formation. Okay, so first so suppose we have an enzyme here, we have a substrate here. So as a first step, what will happen? The substrate will be coming. This is the substrate, this is the enzyme. Okay, so substrate will be coming and it will bind with the enzyme, and this will lead to the formation of enzyme substrate complex. So this is the second step. So first enzyme will be binding with the substrate will be binding to a active site. Okay, this is the first one. Okay, first step, substrate will be coming and binding to the active site. So this is the active site of the enzyme, and to this active site, onto this active site, the substrate will be binding. Second, the formation of enzyme substrate complex formation. The substrate enzyme complex will be formed. So this will be the second step, and third step will be the chemical bonds of the substrate will be broken. Okay, so within the substrate the chemical bonds will be broken and the products will be formed. So once the product is formed, the product will be released from the enzyme, and now the enzyme is again ready for the another catalytic cycle. So after the substrate enzyme complex formation, what will happen? Release of products. So this will be the third step, and now the free enzyme ready to bind with another substrate in the another catalytic cycle. So this will be the order. So first will be E, second A. So in which option you have? EA. So only in the third option you have this. Okay, so third option will be the answer. So the last question. So we have already come to the last part of the biology discussion. So the last question is, mitochondria and chloroplast, both double membrane bound organelles. So what are the double membrane bound organelles in our body? Mitochondria, chloroplast, and then the nucleus. So these are the three double membrane bound organelles. So out of which the two are given, mitochondria and chloroplast. So this is true. So the statement 1 is true. Statement 2, inner membrane of mitochondria is relatively less permeable as compared to the chloroplast. This is wrong because both are selectively permeable, both are equally permeable. So we cannot say one is less permeable, one is more permeable, because both are equally selectively permeable. Okay, so first one is true, statement 2 is false. So in which option you have? Statement 1 is correct, but statement 2 is incorrect. So first option will be your answer. Okay, so with this we have completed the biology discussion, and if we analyze what we can infer from this biology part, all the questions were very easy. All the questions are from NCERT only, so you can without any doubt you can stick to the NCERT, and NCERT plays a very important role in this NEET exam. Okay, so because this is the proof, this is the evidence, because all the questions, whatever the questions we have discussed, everything will be from the everything was from the NCERT. Line to line NCERT questions were given. Okay, so next will be the next subject will be discussed, so that will be the chemistry. So I'm handing over this to the chemistry faculties. So thank you.
Okay, this is okay. Okay, ఇది down. ఇప్పుడు ఇది ఉత్తతో వచ్చేస్తుంది కదా. ఓకే, రైట్. Hello viewers, I'm Dr. Kishore, chemistry faculty of residence. So if you come to that chemistry part, question number 51. What is question is there, sir? So your question is asked, number of moles of Faraday is required. That is the question is asked. So your number of moles of Faraday required is equal to number of moles of electrons are transferred. So here in this case -2 Here the question is number of moles of Faraday is required. So here number of moles of Faraday is equal to number of moles of electrons are transferred. Here in this case -2 to 0, it means 2 electrons are transferred, 2 moles of electrons. So 2 Faraday is required for this. 1 mole of MnO4 -2 Mn plus 2. Here oxidation number is +7, here +2. So totally here 5 moles of electron is transferred, is nothing but 5 Faraday is required. Then 1.5 moles of calcium from molten calcium chloride, calcium to calcium chloride, 2 moles of electrons are transferred. So for 1 mole 2 electrons are required, for 1.5 moles 3 Faradays are required. 1 mole of iron oxide to ferrous oxide to ferric oxide. Here iron oxidation number is +2, here iron oxidation number is +3. It means 1 mole of electron is transferred. So here 1 Faraday is required. So first one is 2, first one is second one matching type questions. So B is 4. So answer is Next, go for that question number 52. So, which reaction is not a redox reaction? Redox reaction is nothing but, in that case, one molecule undergoes oxidation and another molecule is undergoes reduction. So, here H2 and chlorine undergoes oxidation and reduction, but in case of second option, barium chloride reacts with sodium sulfate, get converted into barium sulfate and sodium chloride. There is no change in their oxidation number. It is a double displacement reaction. So, answer is second one. If you go for that question number 53. So, intramolecular hydrogen bonding is present in. So, in meta position, there is no possibility of intramolecular hydrogen bonding. In HF, intermolecular hydrogen bondings are present, but in this case only in between oxygen and hydrogen, partial positive charge of hydrogen and partial negative charge of hydrogen. Here intramolecular hydrogen bondings are present. So, our answer is 3. If you go for that question number 54, Paling solution A is, actually Paling solution is a mixture of two solutions, that is Paling solution A and Paling solution B. So, Paling solution A is nothing but, that is aqueous copper sulfate solution, and Paling solution B is nothing but sodium potassium tartarate, that is a Rochelle salt. So, here Paling solution of A is asked. So, answer is aqueous copper sulfate solution. So, that is the right answer. And question number, next question 55. 1 gram of sodium hydroxide was treated with 25 ml of 0.75 molar HCl solution. The mass of sodium hydroxide left unreacted is equal to. If you come to that equation, NaOH plus HCl is gives rise to NaCl plus H2. Moles of NaOH is equal to moles of HCl. So, moles of NaCl. What is given? 1 gram sodium hydroxide, weight by molecular mass. So, 1 by 40, that is equal to moles of HCl is given, that is 25 ml and 0.75. So, 25 into 0.75 by 1000. So, 1 by 40 is nothing but 0.025. We can write it 25 into 10 to the power of minus 3. This value approximately 18.75 into 10 to the power of minus 3. Now, in these two cases, which is having excess, NaOH is excess and HCl is less amount, so it is considered as a limiting reagent. So, reaction takes place according to that limiting reagent. So, number of moles of NaOH left, number of moles of NaOH left, that is 25 into 10 to the power of minus 3 minus 18.75 into 10 to the power of minus 3. So, what is the value is obtained? 6.25 into 10 to the power of minus 3. Now, your weight is asked. The mass of sodium hydroxide left. This is the number of moles of NaOH is left. Now, the weight of NaOH is equal to 6.25 into 10 to the power of minus 3 into 40, that is equal to 250 milligrams. So, answer is 250 mg. This is the right answer. If you go for that next question, that is question number 56. Again, matching type of question is given. sp3, list 1 and list 2 is given. It means molecule is given and their structure is given. So, NH3, sp3 hybridization, but one lone pair of electron is present. So, structure is trigonal pyramidal structure. BrF5, sp3 d2 hybridization, but one lone pair of electron is present. So, structure goes to square pyramidal, and xenon tetrafluoride minus, actually xenon tetrafluoride, that is sp3 d2 hybridization, it is not minus. So, xenon tetrafluoride, sp3 d2 hybridization, two lone pair of electrons are present. So, square planar structure, and SF6 sp3 d2 hybridization, there is no lone pair of electrons on central atom. So, it is a octahedral complexes. So, what is the right answer for this? Here A is 1 and B is fourth one. It means third option is the correct answer. If you go for that next question, question number 57, the E0 value for the Mn plus 3 to Mn plus 2 couple is more positive than that of chromium plus 3, chromium plus 2, and Fe plus 3 get converted into Fe plus 2 due to the change of Mn plus 3. Mn plus 3 absorbs one electron, get converted into Mn plus 2. So, actually manganese atomic number is 25, electronic configuration 3d5 4s2. Now, it is in plus 3 oxidation state. So, electronic configuration is 3d4 Now +2 oxidation state is nothing but 3D5. So what is the change here? D4 to D5 configuration change. So this is the right answer for this question. If you go for that next question, again, match type of question is given. This is one of the easy question. Basic thermodynamics question is given. So isothermal process, that is carried out at constant temperature, and isochoric process, it is carried out at constant volume. Isobaric process is there. So isobaric process is nothing but constant pressure, and adiabatic process is there. There is no heat exchange in between system and surrounding. So what is the correct answer from this? A is 2 and B is 3 and C is 4, and here right answer is second one. Now go for that next question, question number 59. Activation energy of any chemical reaction can be calculated if one knows the value of orientation of reactant molecules during collision, rate constant at two different temperatures, rate constant at standard temperature, and probability of collisions. Correct answer is this one, rate constant at two different temperature. According to that Arrhenius equation, log K1 by K2 is equal to EA by 2.303 into R to 1 by T1 minus 1 by T2. Okay, this is the rate constant of two different temperatures. Okay, rate constant at two different temperatures. Then go for that question number 60. A compound with a molecular formula C6H14 has two tertiary carbons. Its IUPAC name is. So it is a saturated compound C6H14. If you go for that first compound, 2,3-dimethyl butane. Butane is given. So a compound with a molecular formula C6H14 has tertiary carbons, two tertiary carbons. So directly the first option is the correct answer. These are the two tertiary carbon atoms. So the answer is 2,3-dimethyl butane. Now go for that next question. Question number 61. Spin-only magnetic momentum is same for which of the following ions? Titanium +3 is there. Scandium, titanium. Titanium atomic number is 22. Electronic configuration 3d2 4s2. Now it is in +3 oxidation state. 2 electrons from this, 1 electron from this. So only one unpaired electron is present. Then manganese +2 is there. So manganese +2 atomic number 25. 3d5 4s2. 2 electrons are lost. Then it is having 3d5 electronic configuration. That is 5 unpaired electrons are present. Then chromium +2. Chromium +2, 3d5 4s1. So 3d5 4s1. 1 electron from this, another electron from this. It is having 4 unpaired electrons. Now go for that scandium. Scandium +3 is there. A, B, A, C, B, E. One more is given, I think. That is Fe +2 is given. Fe +2 is also given. So scandium +3 is nothing but, what is the electronic configuration? Atomic number is 21, 3d1, 4s2. So +3 oxidation state. So number of unpaired electrons are 0 for this. And one more is there, Fe +2. Fe +2 is, atomic number is 26 for that iron, 3d6, 4s2. So after the losing of 2 electrons, it is having 6 electrons. 1, 2, 3, 4, 5. So here also 4 unpaired electrons are there. So which is having equal number of unpaired electrons? Chromium +2 and another one is iron +2. So here actually one of the option was missed, and option 3 according to that question paper, option 3, B and D is only right answer. Next go for that another question. Question number 62. Arrange the following elements in increasing order of their electronegativity. So what are the elements are given? Nitrogen, oxygen, fluorine, carbon, silicon and nitrogen, again nitrogen. So this is not one. So here if you arrange this carbon, silicon, nitrogen, oxygen, fluorine. If we move in a period from left to right, electronegativity values are increases. If we move in group from top to bottom, electronegativity values are decreases. So the correct answer is silicon is less than carbon is less than nitrogen is less than oxygen is less than fluorine. So fluorine is more electronegative and silicon is less electronegative element. Silicon is less than carbon, nitrogen is less than oxygen is less than fluorine. So the correct answer is 3. Next go for that question number 63. Which of the following alcohols reacts instantaneously with Lucas reagent? So actually our EDP operator's mistake is there. Here some options are not given. So here actually according to that tertiary alcohol is given. So that tertiary alcohol reacts with Lucas reagent. You know that what is the Lucas reagent? That is a mixture of concentrated HCl and anhydrous ZnCl2. HCl concentrated and anhydrous ZnCl2. So here immediately turbidity is formed. So it is highly reactive. This is takes place according to that SN1 reaction mechanism also. So turbidity is immediately formed. So tertiary alcohol is the answer. Now go for that question number 64. Given below are two statements. Statement 1, both hexamine cobalt and hexafluorocobalt complexes are octahedral but differ in their magnetic behavior. Okay. Statement 2, hexamine cobalt complexes is diamagnetic whereas hexafluorocobalt complexes paramagnetic nature. So in the light of the above statement, choose the correct answer from the options given below. In both the cases, cobalt oxidation number is 3. So cobalt in first statement 1, in statement 1 and in statement 2, in both the cases cobalt oxidation number is +3. +3 is nothing but cobalt atomic number is 27. So 3d7 4s2. If we lost 3 electrons, it is having 3d6 electrons. Here also it is having 3d6 electrons. But here ammonia is act as a strong field ligand. Ammonia is act as a strong field ligand. If you split it in octahedral complexes T2G and EG. So 6 electrons are due to that strong field ligand, 6 electrons are arranged in T2G orbital. There is no unpaired electrons. No unpaired electrons, it is a diamagnetic nature. No unpaired electrons, so it is a diamagnetic nature. Whereas here fluorine is there. Fluorine is a weak field ligand. If you arrange this, due to that weak field ligand, all the orbitals are filled by each one electron. 1, 2, 3, 4, 5, 6. Here how many total number of unpaired electrons are there? 4 unpaired electrons are there. Due to the presence of 4 unpaired electron, it shows paramagnetic nature. So here diamagnetic and paramagnetic both are the octahedral complexes. Statement 1 is true and statement 2 is also true. So what is the answer? Statement 1 and statement 2 both are true. So answer is 3. Go for that next question. Question number 65. Given below are two statements. Statement 1, the boiling point of hydrides of group 16 elements follow the order H2O is greater than H2TE is greater than H2SE is greater than H2S. Okay, statement 2, on the basis of molecular mass, H2O is expected to have lower boiling point than the other members of the group, but due to the presence of extensive hydrogen bonding in H2O, it has higher boiling point. In the light of the above statement, choose the correct answer from the option given below. So as the molecular mass increases, boiling point is also increases. So in every group from top to bottom for that hydrides, molecular mass is increases, then boiling point obviously increases. But here H2O having special case, H2O having hydrogen bonding, intermolecular hydrogen bonding. Due to the presence of intermolecular hydrogen bonding, H2O having highest boiling point than this one. So statement 1 is also correct and statement is also 2 is also correct. So both statement 1 and statement 2 are correct. This is next, go for the next question. Question number 66. Again matching type question is given. So here ML. So ML is indicates that orientation of orbitals. MS is nothing but orientation of spin of electrons. L azimuthal quantum number, shape of the orbitals, and N is nothing but size of orbitals. So A is 3 and B is 4. So answer is 1. So the correct answer is 1 for this. Question number 67. Question number 67. Here, this is the compound is there, get converted into ketones. So what is the suitable reagent? List 1 and list 2 is given. List 1 is reaction and list 2 is reagent and conditions. So here, the first double bond is undergoes breaking. That is due to that ozonolysis. If we apply that ozonolysis reaction, then 2 moles of ketones we can obtain. So this is the answer. And this is Friedel-Crafts acylation. So when benzyl chloride reacts with benzene in presence of anhydrous AlCl3, Friedel-Crafts acylation is takes place. So then after, what is the compound we can obtain? Here biphenyl methanone we can obtain. This is the compound. So here for this Friedel-Crafts acylation 2, and this is alcoholic group is there and here keto group is there. So oxidizing agent is required. So chromium trioxide is one of the oxidizing agent for the conversion of secondary alcohol to ketones. And D, alkyl group is there. That alkyl group is replaced by carboxylic acid, salts of carboxylic acid. It indicates that KMnO4 when it is reacts with entire the carbon chain which is having at least one alpha hydrogen, they undergoes oxidation reaction and COOH is converted. After that KOH is there. So COOH reacts with KOH, they get converted into COOK. So this is that. So what is the answer for this? A is 4 and B is 1. So both are, and C is 2. So correct answer is 1. So question number 67, correct answer is 1. Then go for that, identify the correct reagent that would bring about the following transformations. Now your aldehyde group is there, your double bond is there. So aldehyde group is nothing but when it is alcohol undergoes oxidation to form aldehyde. So primary alcohol is required. So for the conversion of this one, CH2 double bond. Okay. CH2 now here CH2 CH double bond CH2. So when it is reacts with BH3 H2O2 in alkali medium, primary alcohol we can obtain. If you go through that mechanism we can obtain. So CH2 CH2OH we can obtain. So this when it is get converted into aldehyde group, so the best one is PCC. So answer is fourth one. When it is reacts with PCC, then alcoholic group get converted into aldehyde group, CHO group. CH2OH group get converted into CHO group. So question number 68, answer is fourth one. Then go for that next question. That is question number 69. The reagent with which glucose does not react to give the corresponding test or product R. So glucose reacts with Tollens reagent, but it does not give Skiff's reagent test. It reacts with HCl and it reacts with hydroxylamine also, but it does not react with sodium bisulfite. So answer is B and E. So correct answer is 1. Question number 69. Correct answer is 1. Now go for that question number 70. Choose the correct answer from the options given below. It's a very easy question. And here ethane, in between the two carbon atom, one sigma bond is there. So one sigma bond. So this is the third option. Ethene, in between two carbon atom, one sigma bond and another one is pi bond is there. So for this option is 4. And carbon molecule that is having 2, it is not a sigma bond, 2 are the pi bonds. We can explain according to that molecular orbital theory. 2 pi bonds are there, so it is option is. And ethine, in between the two carbon atom, triple bond is there, one is sigma bond and the remaining 2 are the pi bond. So the correct answer is one is fourth one, and ethene second one is third one, and B is the fourth one. C is 2 and D is a 1 sigma bond and 2 bond. So correct answer is fourth one. Now the remaining questions are followed by Dayakar sir.
Good evening to Resonance live students. Myself Dayakar. Today we are going to discussing NEET 2024 paper chemistry. So you seen that paper already feedback. Most of the questions is from NCERT, directly from NCERT questions. If you are reading NCERT, basically you can answer every question. So you will see those questions. This is 71 question. So among group 16 elements, which one does not show +2 oxidation state? Basically, metals do not show negative oxidation state. Okay, here negative oxidation state which is not shown. Basically, metals do not show. Down the group, metallic character can increases. Then obviously here given metal is polonium. So it doesn't show negative oxidation state. We will see next question. For the reaction 2A gives rise B plus C. For this reaction they given Kc value 4 into 10 power minus 3 at a given time. The composition of reaction mixture is, here they given at equilibrium. Concentrations of A, B, C all are equal. So how to find? You can see option they are asking, they are expecting direction of the reaction. So we need to find QC value. If you know QC value, then I can compare with Kc, then I can say which direction. Got it? Now we can see here QC also same like Kc expression. So you can write product here, concentration of B, C, and reactant concentration is A here, A square. But all the concentration they given equal. Then you can use the concentration of B and C here everything. So you can write 2 into 10 power minus 3. Next, again C concentration also same, 2 into 10 power minus 3. Then followed by A concentration also same, but A square they given. I can write 2 into 10 power minus 3 into 2 into 10 power minus 3. Then you can cancel all the values. Then obviously I can get here 1. So this is QC equal to 1. So but Kc value is 4 into 10 power minus 3. QC is greater than Kc. It indicate This reaction is backward reaction. So which option is matching here? So reaction has a tendency to go backward direction. This is the answer. Next, we can move next question. Which plot of ln k versus 1 by t is constant with Arrhenius equation? So basically you can see here they are given with respect to ln k versus 1 by t. It basically Arrhenius equation. It is directly from NCERT book. We can get answer very easily. So you can check it here. So we know Arrhenius equation, k equal to a into e power minus ea by rt. We can apply ln k equal to ln a minus ea by r into 1 by t. We can write this is y axis, this you can take here intercept, and this is mx. So slope is negative. So which is going to show here? Slope only one graph, that is 2. Got it? So direct answer is second one. This is from directly NCERT book. Next question. In which of the following equilibrium Kp and Kc are not equal? How to find out? We know very well Kp Kc relations. Kp equal to Kc into RT power delta Ng. Ng means only gas state. So you can check it here, wherever delta Ng is not equal to 0, then you can apply perfectly. Delta Ng not equal to 0, then obviously Kp and Kc not equal to 0. This is the relation, then you can find out. So in this case, first case, delta Ng 0 because two reactants, two products, delta Ng nothing but number of products minus number of reactants in the case of gas state. So 2 minus 2. So you can apply similarly for remaining cases also. But in the case of C, third option, this is 2 minus 1. So 1, delta Ng not equal to 0, then obviously Kp and Kc not equal to 0. Got it? So we can move next question. Given below are two statements. The boiling point of 3 isomeric pentane follow the order n-pentane, isopentane, neopentane. It's correct order only. Next, when branching increases, the molecule attains a shape of sphere. This results in the smaller surface area for contact, due to which the intermolecular forces between the spherical molecules are weak, thereby lowering the boiling point. Second statement also correct and first statement also correct, so both are true. So which option is matching? This is both statement 1 and 2, both are correct. So we can find the answer is 3. Next question. The compound that will undergo SN1 reaction with fastest rate. I hope you know that one. SN1 reaction follows intermediate is basically carbocation. So which carbocation is more stable? Which carbocation is more stable? We can find out after removing the halogen. Obviously it won't remove the halogen because it attaches to directly benzene ring. And here after removing, I can get here secondary and benzylic carbocation, and it is primary. We can find out this is secondary, but secondary benzylic is more stable, so obviously it is more reactive towards SN1 reaction. So this basically depends upon stability of carbocation. I hope you know that one. 3, 2, 1. This is the stability of carbocation, and it is secondary and benzylic. That is the reason second I am choosing instead of 4. Next question. The energy of an electron, the ground state n equal to 1 for helium plus is x joules. Then that for an electron n equal to state for beryllium 3 plus and in joules is. So we can check it here. So basic equation or basic formula, you know that one from Bohr's model, energy, how to find out by using equation. So I can write here En equal to minus 13.6 into Z square by N square in terms of electron volt per atom. Or you can choose by converting into joules. 1 electron volt equal to 1.6 into 10 power minus 19 joules. By applying that formula also you can convert into joules. But anyway, basic value they are given x joules. So I am going to be converting into here E directly proportional to En. I can write here Z square by N square. You can apply this formula E1 by E2 equal to Z1 square by Z2 square into N2 square by N1 square. Then you can apply this formula, you can get direct answer. So you can see E1 anyway. How much they are given E1? X. You can use that one. Then E2 we don't know. Then apply here Z1. First one, this is helium atom Z1, that is 2. 2 square nothing but 2 into 2 by beryllium. I think atomic number 4. I can write 4 into 4. Again N2 square. N2 nothing but second shell they given. Then obviously you can write 2 into 2. Again N1 they given for helium. Then you can write 1 into 1. So 4 cancel. Again 4 going to be cancelled. Then E2 equal to minus x. If they are using units joules, you can write in terms of joules. This is your answer. So where is there matching that option? Option 3 is matching. Option 3 is matching. Got it? Next question. So in which of the following process entropy increases? Entropy nothing but disorderness. Entropy nothing but disorderness. Wherever disorderness increases, then that cases entropy also increases. You can check it. Basically gases have much more disorderness. Next liquids. Next solids. Who is converting solid to liquid and liquid to gas? Those cases entropy going to be increases. Or we know already recently we done previous question, delta Ng which is positive for those cases also delta S going to be positive and it is going to be increases. Positive and increases. Delta S going to be positive and increases. Got it? So then which case you can find out here? Liquid evaporates to vapor. It is nothing but liquid to gas. So in this case entropy going to be increases. Temperature of crystalline solid lowered from 130 to 0 Kelvin. Now temperature decreases means disorderness also going to be decreases. Got it? Next you can apply here. So in this case may not be possible. Got it? Next you can check it here. Second case, solid to gas. So delta Ng obviously positive here. Then you can think this also positive. And Cl to convert into 2 moles of gas state. Here also delta Ng positive. Then where is option? A, C, D. Option 1. So this is the correct answer we are expecting. Next. On heating, some solid substances changes from solid to vapor state without passing through liquid state. The technique used for the purification of such solid substances based on the above principle is known as. So you can find out one word. This is hint for us. Solid substances. Where they given? Solid substances changes from solid to vapor. This is nothing but sublimation. We know only one option, that is 4, sublimation. Next. Match the following. This is based on isomerism, structural isomerism in coordination compounds. We can identify same example they given in NCERT book also. There is no difference. There is no new question also, direct examples. By looking NO2 you can say it is emergent ligand. It can show linkage isomerism. You can find A for 2. This also direct example from NCERT, SO4 and Br. This can show ionization isomerism. Then where is that look? You can find out ionization isomerism. This is for 2, 3. This is 3. Next, this is both 2 coordination complexes they are given together. Then we know only one that is isomerism, coordination isomerism. Then last one with respect to water involving and solid isomerism. You can compare here. Then match this one. Which is the correct answer suitable here? You can find here first one. D is 1. Here which options here? 3 and 2. D is 1. Next, you can find C. C is coordination isomerism. C is 4. And I think we will get here D1, C4 is only one option, 3. Next. Given below are two statements. Statement 1, aniline does not undergo Friedel-Crafts alkylation reaction. Aniline does not undergo Friedel-Crafts alkylation reaction. So we know that one, basically we are using Friedel-Crafts alkylation reaction is, catalyst is anhydrous AlCl3. It is nothing but Lewis acid. When you are using aniline, it is Lewis base. Both they can react. I think acid base reactions is much more fast compared to any other reactions. Then obviously first reaction is preferred to this one. Instead of act as catalyst, it can takes place, it is going to be takes place acid base reaction. That is the reason they given. Aniline does not undergo Friedel-Crafts reaction. This is correct statement. Aniline cannot be prepared through Gabriel thalomite synthesis. Especially this is using SN2 reaction and are only aliphatic amines. Aliphatic amines. When you are using aliphatic amines, we can prepare this is Gabriel thalomite synthesis by using this method aliphatic amines, but here they are given aromatic, so obviously we can't use this method for aromatic amines. So both are correct statements. Then you can use this as answer is 3. Arrange the following elements in increasing order of first ionization enthalpy: lithium, beryllium, boron, carbon, nitrogen. Choose the correct answer from the option given below. This question also directly from our resonance material and NCERT. Okay, you can see that is first ionization energy. We can find out across the period. First ionization energy increases. First arrange the elements across the period. These are elements: lithium, so you can use here lithium, next beryllium, boron, carbon, nitrogen. But beryllium is the more energy than boron. Then you know that one, this is 2s2, so fulfilled with respect to s orbital, difficult to remove electron compared to p orbital. So we can arrange that one. Lithium is the least one. Which option that is? So every option is least, and nitrogen is the next one, highest that is. So nitrogen also highest. Here we can eliminate the option. Then after that, compared to boron, which is highest? Beryllium. Which is highest? Beryllium is highest compared to boron. So lithium, boron, beryllium, carbon, nitrogen is the correct answer. Right. Next, the highest number of helium atoms is in. So more number of atoms means more number of moles. That is a basic idea. Number of atoms equal to moles into Avogadro number. You can find which option is the highest number of moles. 4 grams nothing but. We know that one number of moles formula, given weight by molecular weight. Here you can get 4 by 4, then obviously 1 mole. This also at STP conditions. This is supposed to be anyway 22.7 they given. So we'll get here also 1 mole. Next, you can find out 4 moles. This is the highest number of moles. Here 4U. This is the given unified mass. That is the least number, that is number of atoms, because only 4, that means 1 atom that is. But here 4 moles, 4 into Avogadro number. This is the highest number of atoms in given options. So you can choose your answer is as a 3. It is basic question from mole concept. You can find here most stable carbocation among the following. Stability of carbocations we know very well. Tertiary, secondary, primary. You can find which is the tertiary carbocation here, that's your answer. So which is the tertiary carbocation here? Only one, that is second option. So next question we can see here. The Henry's law constant KH values of three gases A, B, C in water are 144 and 2 into 10 power minus 5 and 35 kilobar respectively. The solubility of these gases in water follow the order. Basically we know that one solubility according to Henry's law, solubility of any gas, solubility of gas inversely proportional to KH value. So you can say now which is the highest KH value, that is the lowest solubility. Now you can find out here which is the highest KH value, 145, that is the highest, means that is the least soluble. But B have 2 into 10 power minus 5, that is the highest soluble. B is the highest, A is the least. Which option is matching? B is highest. So here, and A is the least one. So we are expecting answer is 4. Next question. A complex X contains 32% of A and 20% of B and remaining percentage of C. Then the empirical formula of X is. So this is also again some basic concepts of chemistry. Basic one, empirical formula we know that one by using basic calculations we can find out A, B, C. A have percentage here, how much they given? 32. And B is 20. And remaining is, these two are 52, so remaining is 100 minus 52, nothing but 48. So how to calculate we know very well, percentage by its atomic mass. So percentage how much here they given? 32. 32 by its atomic mass how much they provided? So 64. So 64 we can use. This is 20 by again mass is 40 and here 48 again mass is 32. So I can get here by solving 1 by 2 nothing but 0.5, 20 by 40 nothing but 1 by 2, this also 0.5 and this is 3 by 2 will get. So least value is 0.5. I can divide with 0.5 again all the values, all three values. Then we can get here 0.5 by 0.5 equal to 1. Here 0.5 by 0.5 is also equal to 1. And 3 by 2. Nothing but this is 1.5. So you can write here 1.5 by again 0.5. I can get here 3. So A1, B1, C3, which option is matching? This is the fourth option. This is the basic one. Next. The products A and obtained in the following reactions respectively. You can get here AB. This is the basically AB obtained in the following reactions respectively. So here this is the direct question from our NCERT preparation of alkyl halides. Direct question, nothing to change. Direct from NCERT. PCl3 when reacting with 3 moles of ROH, we can get here H3PO3. When they are reacting PCl5 with alcohol, replacing OH with respect to Cl, so this is POCl3. That's all. So where the option is matching, you can find A is H3PO3 and POCl3. You can find option 2. Next, the plot of osmotic pressure pi versus concentration. You can check it the units also. That's by using units you can find very easily. For a solution gives straight line with slope. This also liter bar mole inverse. The temperature at which the osmotic pressure measurement is done is. So this is colligative properties, osmotic pressure. This is the topic is solution, we know very well. So basic formula they are applying here. So pi equal to CRT. C nothing but mole per liter. They are drawing graph C. Okay, concentration. So y equal to this we can take x. This is m slope. So we can draw that pi versus C. This straight line obviously. slope going to be RT. Its value they mention here 25.73. You can use units also here they given liter bar mole inverse, but they are expecting answer is temperature. What is the temperature? How to find temperature? By using this basic relation T equal to 25.73 by R. So R value they given, we can substitute here 25.73 by, what is R value? They provided 0.083. By solving this one, we can get temperature. Which unit? Kelvin. We are going to get Kelvin. This is going to be unit is in Kelvin. So approximately this is 310 Kelvins will get. But they given options in degree centigrade. We need to convert into degree centigrade. So temperature in degree centigrade. Basically you know that one. 310 minus 273.15 Kelvin. It's approximately 36.85 will get in degree centigrade. But near option is only one, that is 3. You can keep the answer. Got it? So next question you can see. For the given reaction, this is a KMnO4 oxidation, KMnO4 with respect to H plus. So it is strong oxidizing agent. Obviously, this part is become acid only. So which option is become that is which is a suitable option? Only one, that is fourth. Only one option. So next teacher going to be continue this one. Our expert chemistry expert Srinivas sir.
Hello students, this is Srinivas, Resonance faculty, right? We are going to be discuss 19th question. Here what is the statement 1 given? It is based on statements. First one, they given statement 1, right? CONH3 taken 6 plus 3 is a homoleptic complex, whereas CONH3 Cl2, actually it's given wrong, it's NH3 4. NH3 taken 4. Okay, is a heteroleptic complex. Statement 2, complex CONH3 taken 6 plus 3 has only one kind of ligand, but CONH3 taken 4 Cl2 plus has one more than one kind of ligand. Yes, statement 1 is right. As you know that if a complex having the only one kind of ligands, only for example you can take the given compound is what? CONH3 taken 6 plus 3. This complex having the only one kind of ligand, so that it is called as what? Homoleptic complex. So the given statement is what? CONH3 taken 6 plus 3 is a homoleptic complex. That statement is true. And they given one more complex, CONH3 taken 4 Cl2 plus is the heteroleptic complex, as it having the right, as this compound has the two kinds of ligands. Here you people can observe two kinds of ligands. One is NH3, one is Cl minus I, and two kinds of ligands are present in the compound. It is called as what? Heteroleptic complex, right? Right? Statement 1 is right, statement 2 also right. And what is the correct option? Statement 1 is true, but statement 2, this is wrong option. Statement 1 is false, but statement 2 is true, right? That is also a wrong option. And third one is the correct option. Both statement 1 and statement 2 both are true, right? This is the basic question they asked from the complex compounds. And question number right. During the preparation of more salt solution, which of the following acid is added to prevent hydrolysis of Fe+2. This is very basic question. Has Fe+2 to prevent the oxidation of Fe+2 into Fe+3. Generally, we had the what nana dilute sulfuric acid. Dilute sulfuric acid. Then answer is what? 2. Right? And question number 92. Identify the correct answers. Dipole moment of NF3 is greater than the NH3, right? Write the structure of NF3. Right, this is the structure of NF3. Right, here this is the momentum. Right, this is the momentum. Here this is the momentum. Here we have the lone pair of electrons, and this is the momentum. Here one momentum going to be cancelled. Sir, right, here when you take the NH3, here this is the momentum, and here we have H, here we have H, here we have H, and all the momentums are get added. All the vectors are get added. Therefore, mu of NH3 is greater than mu of NF3. What is the given statement? Dipole moment of NF3 is greater than that of NH3. That is the wrong option, obviously. And what is the second option they given? See here. What is the second option they given? 3 canonical forms can be drawn for CO3 minus 2. Yes? Is it right? Let us see. Right? What is the CO3 minus 2? This is CO3 minus 2. Here, these negative charge delocalized here. These pi electrons are delocalized here. Right? Here we get the CO minus. Here O minus. Here we get the double bond O. And these electrons are shifted here. These electrons are shifted here. Then what you get, Nana? Another resonating structure. And here we get the double bond. Here we get the O minus. Then we get the this one. How many canonical structure are possible with CO3 minus 2? This is 1. This is 2. This is what? 3. 3 canonical structures are possible with CO3 minus 2. So, option 2 is the correct. And next one. Let's move into next one. Given below are certain cations using inorganic qualitative analysis. They given the question from the qualitative analysis. Arrange them in the increasing group number from 0 to 4. Right now? Right? Observe here. Here Al plus 3. Al plus 3 belongs to third group. Okay na? Right? Al plus 3 belongs to third group, and next they given the what nana? Cu plus 2. Cu plus 2 belongs to second group. That means Al plus 3 is the what nana? This is second one. We get the, this first we have to get the B, next we have to get the A, then no need to check all the options. We can check the third option and fourth option, then we can eliminate the 1 and 2, then we get the what option? Third option is the correct one, right? Next we move to 94th question. So this is very, very basic question. They asked from the organic chemistry, right? And NCERT question. Let us see. Here we have CH3 and we have what nana? CH2, CH2I, propyl iodide treated with what? NaCN, right? What happened? Substitution takes place. CH3, CH2, CH2, CN, cyanide is formed. They treated in the partial hydrolysis in the presence of base. So when it undergoes the partial hydrolysis, what you get nana? CH3, CH2, CONH2. Acid amide is formed. Then they treated with NaOH, Br2. This is very famous named reaction, Hoffman bromamide reaction. Hoffman bromamide reaction. Right? What is the answer? Very simply, you remove this one, then you get the answer. CH3, CH2, CH2, NH2. What is our answer? It is propylamine. Answer is propylamine. Third option is correct one, right? Next we move to The rate of reaction, 95 question. The rate of reaction quadruples when temperature changes from 27 degrees Celsius to 57 degrees Celsius. What they asked, Nana, calculate the energy of activation. Right now, we have to calculate the EA. They given the T1. T1 is the what, Nana? 27 degrees Celsius. Then 300 Kelvin. And they given the T2. What is that T2 they given? 57 degrees Celsius. What is that? 320 Kelvin. Then what we have to calculate EA. And what they said? K1 equals to K. K2 is quadruple. K2 equals to what, Nana? 4K. What is the equation we have? Log K2 by K1 equals to EA by 2.303 R 1 by T1 minus 1 by T2. Right now, what we have to find out? EA. EA is equals to log K2 by K1 into 2.303 R. Okay. Then what you get? T1, T2 by, okay, T2 minus T1, right? Solve it. EA is equals to log K2 by K1. What is the K2? 4K. Because what they mentioned, the rate of the reaction quadruples, right? Then 4 times they increases. Then 4K by K, KK get cancelled, log 4 into 2.303. Answers given in kilojoules. That means R value you should be taken 8.314. What is the T1? T1 is the 300 into 320 by T2 minus T1, 20. You have to solve it. Then you get the answer 38.04 kilojoules per mole. 38.04 kilojoules per mole is the answer. At what is the equation we have to use? Log K2 by K1 equals to EA by 2.303 R and 1 by T1 minus 1 by T2. That is the rate equation we have. And what is the statement? You observe the rate of reaction is quadruples, right? That means rate of reaction they increased by the 4 times. Then what is the activation energy? We have to find out by using this equation. This is a very simple question, right? Right. Next one. Consider the following reaction in a sealed vessel at equilibrium with concentration of N2 and O2 and NO they given, and they given an equilibrium equation. Then they given how many moles of NO taken in the closed vessel, and they asked for the what, Nana? Alpha. They asked for the alpha. It's a very simple question, right? Right, 2NO is equilibrium with what? N2 gas plus O2 gas. Right, initially how many moles of NO is present in the closed vessel? 0.1 moles. Right, this is 0 moles, 0 moles at T is equals to 0. At T is equals to T equilibrium, what happened? It will become 0.1 minus X. It will become what? X by 2. It will become X by 2. Then what is your object? You have to find out the X. That is nothing but degree of dissociation. Right? So for this you have to write the Kc. Kc equals to what? Concentration of N2 into concentration of O2 at equilibrium by what you get? Concentration of NO square. Okay, na? Right? Then you solve Kc equals to X by 2 into X by 2, okay, by 0.1 minus X whole square is equals to what you get if you solve this 3 into 10 power minus 3, okay, 3 into 10 power minus 3, what they given? 4.2 into 10 power, sorry, I will write here. Yeah, I will write somewhere else. 3 into, yeah, just wait. Right, Yeah, right, I will solve here. It will get 3 into 10 power minus 3 into 4 into 10 power minus 3 by 2.8 into 10 power minus 3 whole square. This one get cancelled, and here you get the 1.6, and then solve the x. You get the x almost equals to what? What is the answer you get? This one. This is the way you can use, right? By using this way, you can find out the what degree of dissociation. And the work done during the reversible isothermal direct equation, reversible the isothermal expansion of 1 mole of hydrogen gas at 25 degrees Celsius, pressure of 20 atmosphere to 10 atmosphere. They given P2, P1 and all 1 mole. The W is equals to minus 2.303 RT, NRT log P2 by P1. Okay, W is equals to minus 2.303. Just wait. P1 by P2, right? Right, 2.303, N is 1, R is 2, and temperature they given what? 25 degrees Celsius, 298 log 20 by 10. If you solve, you get the minus 413.14 calories. It's very simple question. You can use the logic. Here negative term is there. In the any other in 3 options there is no negative term. Then you can choose option 4, right? And next question, right? Here we have what is the given OH substrate is this one. We are treated with PBr3 and SN2 mechanism. This OH is substituted by Br, and what you get? Here we get Br. Okay. Then what is the reagent they used? Alcoholic OH delta, and elimination takes place, right? And set job product is the major product. This is beta hydrogen. This is beta hydrogen. You should give the major product as the answer. Then what is the answer you get? Set job product is the major one. Sorry. Right? Set job product is the major product. Then what is the answer you have to give? Option this one. Okay. Right? Next one. Last question. The pair of lanthanides which are diamagnetic. Diamagnetic means they should not having the unpaired electrons. They should not having the unpaired electrons. If you observe cerium 58 and ytterbium what nana 70. What is the electronic configuration? Xenon 4f1 and 5d1 6s2. What is the ytterbium electronic configuration? 4f14 6s2. They given cerium plus 4. Then what is the electronic configuration? Xenon 4f0 5d0 and 6h0. There is no unpaired electrons, so this is diamagnetic. And they given ytterbium plus 2. Then what is 4f14? This one, right? Xenon 4f14. Here also no unpaired electrons. This is diamagnetic. This is also diamagnetic. And the option is what? 3. Right? That's it. Okay. Next, next they are going to be discuss physics. Right? Thank you. Yeah.
Hello students. So I hope so you have done very good in the NEET examination. Physics paper was, as we are expecting, it's a easy to moderate side. And if what we had discussing at that time also, that most of the questions are either from our grant tests or from our materials itself. So those students who has gone through the grant test seriously, almost 100% of the physics questions they have done. So let's start with the discussion part. First question is, a tightly wound 100 tons of coils, radius is given, current is given. Magnitude of magnetic field at the center of the coil they are asking, and value of mu they have already given. So let's start here. Just wait a minute. Just a minute. So first question is, coil is given and they are asking just with the magnetic field at the center of the coil. Marker is not working. So here a coil is given and the number of turns is given as 100 and they are asking at the center of the coil. So we have a formula for this that we have already explained in the class also. B is equal to mu naught I by 2R into number of turns. Value of mu naught is given, that's a 4 pi into 10 power minus 7. Value of current is also given, that is a 7 ampere. 2 radius is also given as a 10 centimeter. N turns is given. If you will solve this question, you will get the answer easily. So it's the easiest question for us. Now coming on the question number 2. See, this is again the theoretical question. We have explained this question and n number of times we have discussed this question, that if it is a diamagnetic, what we know about the diamagnetic, that it does not likes the magnetic field, it repels there. So which one will be the answer? This one. Ferromagnetic, which loves a lot in magnetic field. So which one? That's a very susceptibility will be very large as compared to one. And paramagnetic, feebly they presence, they likes it. This one. Non-magnetic, this one. So which option will be the answer? Diamagnetic means this one. B. Ferromagnetic. C. Paramagnetic. This one. Non-magnetic. This one. So first answer is B. Second answer is third. So this option will be the correct answer. Now coming on the next question, again the easy question. See here. See, this question we have discussed in detail, that in case of isochoric process, isobaric process, isothermal process, how to find the work done. They have asked in which process where the volume is constant. And if the volume is constant, work done equal to what? P dot dV. So volume is constant, so work done equal to what? In case of isochoric process, work done is 0. Again the easiest question for us. Coming on the next part. Next question is, an unpolarized light beam strikes on a glass surface. Let's take this as a glass surface. Light strikes over here. After this it will pass over here. I, and this is reflected because it's a Weber's angle. We have discussed this thing again in detail. This angle becomes what? 90 degree. And in this part, what is happening? This is the completely polarized. Whenever the reflected light is, it's a completely polarized. And for the refracted lights, it's a partially polarized. So which option will be the answer? Second option will be the correct answer. Now coming on the next part. Question number 5. Now see, again, this is the easiest question for us. NP by NS is equal to Vp by Vs, and they have given how much? 1 by 2. But they have asked here Vp by Vs, or they have asked here Vs by Vp? They have asked here Vs by Vp, and that is equal to becomes what? 2:1. So this is the thing where we should take care during the NEET examinations. Set 1:2, 2:1. They have given this. Both options they have given this one. Now again, we have done good level of question in our class, in our module, but here they have asked 0, 0, AB, 0, 0, 1, 0, 1, 0. So which option is the answer? Not. Simple not is the answer. Now coming on the next question. In a vernier caliper, n+1 divisions of vernier scale coincides with the n divisions of main scale. So what we can say, that n+1 VSD is equal to what? How much? NMSD. So what is the 1 VSD? Sir, 1 VSD is equal to n by n+1 MSD. Now, so what is the least count? So least count we all know very well, that 1 MSD minus 1 VSD. That's equal to 1 MSD minus 1. And in place of VSD, what we can write? N upon n+1 MSD. So this will becomes equal to what? n+1 minus n. So n+1 minus n upon n+1 MSD. Now, so it becomes how much? 1 upon n+1 MSD. And what is the value of MSD they have given? MSD value they have given as 0.1 millimeter. But see here what they asked. They have asked the answer in centimeter. So 1 upon n+1 into 1 by 100 centimeter. So which option is the answer? 1 by n+1 into 100. This question. Now coming on the next question. Next question is maximum elongation. Maximum elongation of a steel wire of length is given. Wires of length is how much? That is a 1 meter. Elastic limit and Young's modulus. So it means the stress is given and Young's modulus is given. We all know this part, that what is the formula of Young's modulus? So that is the stress and divided by strain. And what is the strain? That's a delta L by L. Now in this, see here, they have given this one, they have given this one, they have given this one, they are asking what? Delta L. So delta L is equal to what? Stress divided by Young's modulus into length. Just divide this part. Stress is given as 8 into 10 power 8 divided by 2 into 10 power 11 into length is 1 meter. So how much will be the answer? 4 into 10 power minus 3 is the correct answer. So 4 mm is the correct answer. Now coming on the next question. Question number 9. Again the easiest question for all of us. Easiest question. We have discussed n number of times this more than this level of question. See, what is the acceleration of the system? Acceleration of the system becomes what? So net force upon total mass. How much it is? 2 meter per second square. Correct? And after this, they are asking what? Force exerted by block A on block B. So either we can draw the free body diagram on block A, or we can draw the free body diagram on block B. Anything is fine for us. Now I am drawing the free body diagram on block B. 3 kg, this is the normal force, and acceleration as we have already found, it's a A. Is how much? That's a 2. Now apply the force. Force is equal to mass into acceleration. 2. And equal to how much? That is a 6. Which option is the correct answer? First option. Now coming on the next question, question number 10. In the question they have asked in a monochromatic source of YDSC experiment replaced by white light. What is the difference between the monochromatic? Monochromatic means single wavelength of light. And what is the white light? The combination of all colors. That is the white light. So this is when replaced by the white light, then what happens there? So all white light will merge there here. See this one. So what will happen? This all white light will come over here. All will reach over here. So this becomes what? Here all colors of light will reach. All colors of light will reach. So on this, what will happen? This again the combination of all colors, it becomes the final, the central bright white light fringe surrounded by the few colored fringes will get it. So which option is the correct answer? First option is the correct answer. Now coming on the question number 11. Here they have asked the relation between what? 1 by lambda square and the kinetic energy. So relation, whenever this graphical forms is there, how we are doing this question? We are finding the relation between what? The kinetic energy and lambda square. So how we can find? Sir, one relation what we have discussed, that 1 lambda is equal to h upon root 2m into kinetic energy. H upon root 2m kinetic energy. So squaring on both sides. So squaring or just take the cross multiplication and then square it. 2m kinetic energy is equal to h square by lambda square. So from here what we can say? Energy is equal to h square by 2m into 1 by lambda square. So this is what? Y is equal to mx equation, and y equal to mx equation is what? Straight line passing through the origin. So which option will be the correct answer? Again, this is little better question. It's a good question. Now coming on the next question. See here, equivalent capacitance for this. So as we know this part, this is again what? 2, 2, 2 and 2 balanced with tone. So for us this becomes the useless. So this 2 and this 2 becomes in what? Series. So become how much? 2, 2 becomes series. In series. Two becomes what? Series becomes 1. And this 2 becomes series what? 1. Now what is the equivalent part? 1 and 1. That is in parallel. It becomes what? 1 plus 1 equal to 2. Orally question, this question we can do easily. Coming on the next question. Question number 13 is, strong bar magnet is here and this is moving. We know this part, when if it is moving, so what will happen? This will repel and this A to B side. So here what will generate? It will not, it don't want this to go away. So here the south pole will generate and here the north pole will generate. And here the south pole and here the north pole. It means how it should be? This is only if the current will go like this way. So this way the current will go. So current will go from here to here. That is from A to B. Now coming on this one. This current is here C and D. This is from the inside. This one. In this, here what will happen? The south is moving towards it. So again this will generate what? South, and this becomes what? North. It is when it is possible, if it will be like this way. If it will be like this way, it means what? The current will go like this one. This way. So current from here to here. To D to C. So which option is the correct answer? A to B and D to C. Third option is the correct answer. Now coming on the next question. Here, this is again this question again we have discussed in our class notes. Solar cell. Solar cell graph is what? Where in the fourth quadrant. So yes, they have asked in the fourth quadrant. Yes, it is correct. Now second is the in a reverse biased PN junction. Reverse biased means what? PN. And here is what? Higher putting. This is the P. This one is N. So this becomes what? Reverse biased. So in case of reverse bias, what will happen? The current measured in microampere is due to measure T, charge carriers, No, no, no. This is not because of majority charge carriers. This is because of what? Minority charge carriers. So which of the correct answer? First option is correct and second option is not correct. So A is correct but B is incorrect. So third option is the correct answer. Now coming on the next question. Again, this is the easy question for us. Now here, this is the 90 degree. So this becomes what? Critical angle. So R2 is the critical angle. So what is the R1? We know this formulation that R1 plus C. R1 plus C is equal to what? R1 plus R2 is equal to what? A. And A is how much given? That is the 90 degree. R1 plus C is equal to 90. So what is the R1? 90 minus C. Let's now apply the Snell's law on first surface. What we can write? 1 into sine 30 is equal to mu into sine of R1. And what is the R1? That is the 90 minus C. So half is equal to mu cos C. And what is the critical angle? We all know this part. That's a sine C is equal to 1 by mu. Now just in a triangle, because we need the cos C, sine C 1 by mu, this becomes how much? Root of mu square minus 1. So 1 by 2 is equal to what? Mu into cos is root of mu square minus 1 by mu. Mu will cancel. So squaring on both sides, mu square minus 1 equal to 1 by 4. So mu square equal to 5 by 4. So what is the value of mu? It's of root 5 by 2. Which option is the correct answer? This one. Good question. Because the calculation is required here. Now coming on the next question. Question is here the potential of any at any axial point for which from the center of a dipole. What is the formula here? Sir, Kp cos theta by r square. So in case of dipole, if this is the direction of dipole, here it becomes what? Positive. And this side it becomes what? Negative, because here cos 0 and here is cos 180 degree. So that's why it is written as plus minus this one Kp cos theta by r square. That all the things is given here. If I'll put the value Kp by r square, put the value of K, that is a 9 into 10 power 9. Dipole moment is also given 4 into 10 power minus 6 by r square. What is the r? 2 square. That's a 4. This will cancel. So which option is the correct answer? 9 into 10 power minus 3 plus minus y, that we understood because one is the right side and other is the left side. That's it. Now potential region they have given what? K 2 Kp. This is the mistake what they have done. It's not 2 Kp cos theta by r square. It's a Kp cos theta by r square. So first is true and second is false. This is the correct answer. Next, coming on the question number 17. Again the easiest question, they are asking the moment of inertia perpendicular to the rod. It's what? If the moment of inertia they are asking perpendicular to the rod, this is the axis omega. So what is the answer? I is equal to ml square by 12. I equal to what? ml square by 12 is the correct answer. They have given the value of mass, they have given the value of length, all the things they have already given. After solving this, we'll get the answer as 8.5 centimeter. Or just put the value, we'll get the answer. Coming on question number 18. Terminal voltage of battery, they are asking. EMF they have given the 10 volt. And resistance, internal resistance of the battery also they have given the 1 ohm. External resistance they have given the 4 ohm. So they are asking what is the terminal voltage. And in the case we know, if the battery acts like as a source, then the terminal voltage is what? E minus IR. And when the battery acts as a load, then the terminal voltage is what? E plus IR. So simple thing, what is the current here it will flow? Current here is equal to how much? I is equal to V by R. So V is 10 and 4 plus 1 is 5. It's a 2 ampere. Now, what is the terminal voltage? Delta V is equal to E minus I into internal resistance. So what is the value of E? 10 minus I is how much? 2 and R is how much? 1. So answer is what? 8. So this is the correct answer. Which option is the correct answer? This one. Now, coming on question number 19. Here, they have given this, that transition they have given from 3 to 2, and they are asking the wavelength. We know this part, the value of the equation is 1 by lambda is equal to rz square 1 by n1 square minus 1 by n2 square. N1 is the orbit, inner orbit, and N2 is the outer orbit. Value of Z they have already given, it's a hydrogen, and value of R is what? 1.1 into 10 power 7. Just putting all the equations, values over here, will get the value of lambda. But for the medical students, our suggestion is what? Rather than going by this formula, taking all these calculations, you should use little bit how this simple 1 by lambda, just solving it, this equation, you will get this one. Lambda is equal to n1 square into n2 square upon n2 square minus n1 square into r, 1 by r. And value of 1 by r is what? 912 angstrom. Just putting all the values, easily you will get the answer. Coming on question number 20. C is the velocity of light they have given in the free space, and statements about the photon they are asking. Energy is h nu, yes, it is correct. Now velocity of photon is c, yes, it is correct. Momentum is equal to h nu by c, yes, it is also correct. Both total energy and total momentums are conserved, yes, also it is correct. Photon possesses positive charge, that is not correct. Photon does not have any charges. So which option is the correct? Correct one is A, B, C and D. This one is the correct answer. Now coming on question number 21. See, here is a alpha decay. In this question, here is a alpha decay. So X is there, 82, 290 is there. If there is a alpha decay, it means they are removing here what? Helium, 2, 4. So it becomes, Y becomes what? Y becomes, you are removing the 2, means it becomes 80 and 286. Now from here, they are removing one electron plus. It means what will happen here? Here, neutron, there's a two thing. When the proton will break, this becomes neutron plus electron. So this equation will not, this is what? Beta minus decay. And when the neutron will be there, this becomes what? Proton. No, this plus E plus, beta plus decay, and neutron will break plus beta minus decay. Now here, what will happen here? In case, whenever there is a E plus, it means what will happen? One proton will convert into what? Neutron. So what will happen here? In case of Z, 81 becomes what? 79, and this remains the same. Now in case of there, how again there's a beta minus, this one. Now this neutron will convert into proton. P becomes what? 80, 286. Again Q, again beta minus decay, so it becomes 81, 286. So which option is the correct answer? 81 and 286. This option is the correct answer. Now coming on question number 22. Here displacement is given as 2t minus 1. Under the influence of force, this one. Value of instantaneous power, there is some ambiguity in this question. But let's go as per this question, because of this constant force, this equation cannot be in linear equation, it should be in quadratic equation. But okay, let's assume that because of any other forces are acting there, and because of that displacement is this one, and we are assuming that force and velocity both are parallel to each other, then under this consideration we are solving. So here displacement is given, how much? 2t minus 1, and we know this thing, that ds by dt is what? That is the velocity. So that is equal to what? 2. Now power is equal to, they have asked here what? Power, and power is equal to what? F into V. We are assuming this thing, that force and velocity is parallel to each other. What is the force here? That's a 5. And how much velocity is there? That's 2. So answer is 10. Which option is the correct answer? Third option is the correct answer. Now coming on question number this. This is what? This becomes the A bar. This becomes what? B bar. And here it becomes what? A bar plus B bar bar. So it's what? AND gate is the correct answer. Now coming on question number 24. Mass of the planet is given as 1/10 of the Earth and diameter is half of the Earth. Accretion they are asking. What is the accretion due to gravity? G is equal to Gm by R square, or we can say the D square by 4. That we can write in terms of diameter. Now let's come for this accretion for this other planet. G becomes constant, mass becomes m/10, diameter becomes half. So it means D square by 16. Now just divide it. So G by A is equal to what? Gm by D square. Gm by D square remains, this will cancel, is equal to 10 by 4. So what is the A? 4 into G by 10. So A is equal to 4 into 9.8 by 10. So what will be the answer? Answer will be 3.92 meter per second square is the correct answer. Now coming on question number 25. Question 25 is, atoms are electrically neutral as they contain equal number of positive and negative charge. Yes, it is correct. Because of number of protons and electrons, both are equal. So it's a neutral. Yes, it is correct. Now coming on statement 2. Atoms of each element are stable. Each element are stable. That is not correct. Why it is? Because we have n number of examples. What we have discussed in the radioactivity, that nucleus is not stable, and if the nucleus is not stable in case of uranium, and any other examples are there. So atom, how it can be stable? So saying the atoms of each element are stable is not correct. So which option is the correct answer? Statement 1 is correct, statement 2 is incorrect. Now I request Mohit sir to come and continue from question number 26. Please, sir.
So, hello everyone. We'll continue with question number 26. Sir has already briefed you about that, how this paper was. It was more of a theoretical question with few amount of formula-based questions also. So, part of it has been discussed by Rahul sir. Let's continue with me with question number 26. So, we'll go a little fast, and as per need, if there's a discussion required, extra discussion required, we'll go for it, right? Going for question number 26. A wheel of bullock cart is rolling on a level road. So the word which we want to highlight here is rolling. The moment it's rolling, rolling means we are assuming it's a rolling without slipping, which means the point, this point has contact velocity 0, right? Which means that V is equals to omega R, right? It's rolling, right? Omega is the angular velocity. So continuing with this rule, so this point will have V plus omega R, where omega R is equals to V, which means V plus V, which becomes 2V. So let's go forward. On a level road as shown in figure below, if its linear speed is V in the direction shown, which one of the following option is correct? P and Q are highest and lowest point on the wheel respectively. So P is the highest point, Q is the lowest point. If it is rolling without slipping, obviously this point has to have velocity 0, and this point will be having velocity 2V, right? So point P moves faster than point Q, so we'll go for the option number 4, right? Let's go next. Question number 27. A particle moving with uniform speed in a circular path. Whenever it's a circular path, particle have velocity in a tangential direction and acceleration, which is a compulsory acceleration called centripetal acceleration in the direction of the radius. Radial acceleration we also call it. So when the particle reaches the next point, velocity is again like this, but again acceleration is like this. So at every point, velocity and acceleration is changing its direction. So can we say that velocity and acceleration both are changing? So it's a varying velocity and varying acceleration, as vector quantity depends on value as well as direction. Simple as it. So varying velocity and varying acceleration is the answer. Let's go for next one. 28. A thin flat circular disc of radius 4.5 centimeters placed gently over the surface of water. Something placed over the surface of water, and it's a circular disc. So this is like a circular disc, right? What is the, if the surface tension of water is 0.07, so surface tension is 0.07 Newton per meter, then the excess force required to take it away from the surface is, so F is equals to surface tension into the distance, like the length which is attached with the surface. So that will come out to be 2 pi r as the length, this is length, and surface tension is equals to 0.007. When you solve it, where you put the value of r as 4.5 10 to the power minus 2, as it's in centimeters, so you have to convert it in meter. Solving this will give you 19.8 millinewton. I'm just avoiding the basic calculation. 2 into pi, pi is 3.14, r is 4.5 10 to the power minus 2, 0.07. Going through basic proper calculation, I know the mistake we can do here is calculation mistake only, but if you go through 2 pi r, properly putting the value of r and putting the value of surface tension 0.07, then you'll get the value as the 19.8 millinewton as the answer. Okay, going forward. In a uniform, 29, in a uniform magnetic field of 0.049 Tesla, a magnetic needle performs 20 complete oscillation in 5 seconds. So what is the time period, by the way, first of all? 20 complete oscillation, 20 oscillation requires 5 seconds, and 1 oscillation require 5 by 20 seconds, which comes out to be 1 by 4 seconds, right? So that's the time period. The moment of inertia of the needle is 9.8, so this is like the value of I. If the magnitude of the magnetic moment of the needle is this, matlab we are trying to find the value of m. So the related formula for this is T is equals to 2 pi root over I by mb. I mean, we go through this formula while our discussion in the theory of this chapter of magnetism. So let's plug in the value of T then the value of T is coming out to be 1 by 4, 2 pi root over I. What is the value of I? I is, where is I? Uniform magnetic field of 0. So this is the value of B. Magnetic needle of 20 oscillation, so time period we got it. This is the value of I. So I is 9.8 10 to the power minus 6 by value of M and B. Value of M we need to find, and value of B is given 0.049. So value of M is given, M is need to be found, and value of B is given 0.049. Squaring both sides comes out to be 1 by 16, 4 pi square, 9.8 by M is a 10 to power minus 6, you have to put there, 0.049. Simple calculation. When you solve this, this turns out to be 200 around, and this will be, sorry, this will be 16. This will be 16 because you squared both sides. So 4 ही रहेगा, 4 will be there. Okay. So solving this, yes, solving this 16 into 4 into 200 and taking this m this side, this comes out to be m equals to 1280 pi square into 10 to the power minus 5. After solving this, we'll get this value, right? So that's option number 2. When you solve this, you'll get this. Simple calculation. T is equal to 2 pi root over I by mb, where the value of I is given, B is given, 0.049 is the magnetic field. So you can get the value of m out of this after simple cross multiplication solving, right? Very simple calculation. Going forward. Okay. Going forward, two bodies A and B of same mass undergo completely inelastic one-dimensional collision. So two bodies, one is mass m, same mass, let's go for the same mass. The velocity is v1 and this was at rest initially. After collision, they come for the body A and moves with velocity v1, undergoes complete inelastic, matlab m plus m, they get stick together. When two masses of the same mass goes for inelastic collision, the net mass now becomes 2m. Having a velocity of v2. The body A moves with velocity v1 while body B is at rest before. This is A, this is B. and they stick together, the velocity of system. System means after they stick together. So obviously what we are going for, when there is no external forces are acting, we apply a basic rule of momentum conservation, which means that m into v plus 0, because the velocity is 0, m into 0 is 0. So 2m into v2 is the formula we are using here. M cancels. So v1 by 2 is the value of v2. So what is the next question? The velocity of the system after collision is v2. The ratio of v1 is to v2. So v1 by v2 is v1 by 2. So v1 by 2, 2 goes to numerator. That becomes 2, which is like 2 is to 1. So the option going right here is option 4, which is 2 is to 1. Yes or no? Right? Going forward, this is x equals to 5 sine pi t plus pi by 3. It's an ideal equation of SCHEM. An ideal equation of SCHEM, which means that we can compare this as x equals to a sine omega t plus phi. So omega is like pi, a is like 5. So this is meter, right? So obviously centimeter wala option first of all will cancel. Meter wala option we are thinking. Now the next comma ke baad wala question is we have to find the time period. So what's the value of time period? Time period is 2 pi by omega. 2 pi by omega. Omega is pi. So pi cancel, which means 2 seconds, right? So the option is option 4. The answer for this question 31 number is 4. Let's go for the next question. 32. The quantities which have the same dimension as those of solid angle. Solid angle have no dimension, dimensionless. Going for dimensionless stuff here. Strain, arc has obviously length. Angular speed has a theta by time, so time inverse. Not dimensionless. I'm not going for stress even discussion for it because one of them have dimension. Let's not go for the other option. Going for strain and angle is the best possible option. Strain is, the meaning of strain is deformation per original length. Deformation per original length. It can be deformation per length by length or delta v by v and so on. So this will be dimensionless, right? This will be dimensionless. This will be dimensionless. This will be dimensionless. Going for angle, angle is also dimensional. Angle has unit, which is theta, but no dimension. So strain and angle again is dimensionless, same as solid angle. So we'll go for option number 3. I'm not going further because single option is correct. Let's not discuss even further. Once I know it's strain and angle. Stress is obviously having dimension, which is force by area, which is not dimensionless, so not as the same as solid angle. Let's go forward. So option number 3 is the answer. Okay. I hope things are very comfortable. We are going little quick. I hope things are being absorbed by everybody. Let's go for question number 33. A thin spherical shell is charged by some source. The potential difference between the 2 points C and P. Dear all, the charges are kept on the surface. There is no charge inside. So electric field is on the surface, right? So there is no electric field inside, which means delta V inside is 0. So the V on the surface, I should write it more clearly. Okay, the delta V is 0. So the V on the surface and V inside is the same as it goes for the conductor. The potential on the surface and potential inside is the same. So there's no such change in potential. So the question is potential difference on the C and P. P is on the point on the surface, this is P. This is 0 is the answer, right? Going forward with the next question. A bob is whirled in a horizontal plane. Horizontal plane, but let's not worry about gravity and all. So means of string with A cap plus A cap. Maybe there's something, some error here. Speed of W, omega is the RPM, omega is the angular speed. The tension in the string, this should be the string, is T initially. If speed becomes 2 omega while keeping the same, sad mein nahi hai bhai, it's not sad mein, it's same radius, the tension in the string becomes. So the tension in the string becomes, when it get doubled, so the formula for T is equals to m omega square R. I hope we all remember. The centripetal force required whenever something is going for a circular motion is T is equals to mv square by R or m omega square R. When omega is been replaced by, 2 omega, this becomes 4 m omega square R which becomes 4 times T. Right? The option will be 4 times T. T hona chahiye 47 nahi 40. The answer should be 40, right? Going next. 35. A wire of length L and X resistance 100 ohm is divided into 10 equal parts. So let's say the total resistance was 100 ohm. We divide into 10 equal parts. So each resistance is how much? 10 ohms. Right? Divided into 10 equal parts, 100 by 10. Right? 100 by 10. So resistance offered by each part, it will be 10. First 5 parts are connected in series, while the next 5 parts are connected in parallel. So the series combination will give you combination of the 5 part in series, 5 into 10 or 5, 10, 10 plus 10 plus 10 plus 10 plus 10 will give you 50 ohms. Whereas the parallel combination will be 10 each by 5, you know, 1 by R plus 1 by R plus 1 by R plus 1 by R, R by actually 5, which is like 2 ohms. Sum of them is like the net, which is 52 ohms, because the question is the two combinations are again connected in series. So if first part you got in series, the next part you got in parallel, then you sum up them, which is 52 ohms is the answer, right? Option number 4. 36. The following graph represents the TV curve of an ideal gas, where T is the temperature, V the volume, at 3 pressure P1, P2, P3. So whenever it's a TV graph and given Charles law means T proportional to temperature proportional to volume. So this pressure obviously constant. It's a slope actually. Maybe the graph picture is not very clear. So obviously if something is ideal gas, we can write PV is equals to nRT, right? They mentioned the word ideal gas. We can write PV equals to nRT, differentiating both sides respect to volume. So P, and I'm assuming Charles law is represented by this dotted line. So I'm not assuming that P is variable here for this line, for this line, for this line. So I'm not going for UV rule. I'm taking P as a constant. So I'll write dV by dt is equals to nR dt by, it's a TV graph, huh? Slope of this graph is TV graph. So I should go for okay. So PV differentiating both sides respect to volume. So P dV by V nR dt by dV. dt by dV is the slope of this graph, right? I hope we all understand. When it's a yx graph, dy by dx is the slope of the graph. So dt by dv is the slope of the graph, right? So this is the slope of the graph. This will become 1. So can I write that dt by dv is proportional to p? dt by dv will be p by nr. So I can say dt by dv is proportional to p, right? So when we have more value of slope, means more value of p, lesser value of slope, lesser angle with the x-axis, lesser value of slope means lesser value of p. So we can see the highest slope with p1, then lesser with p2, then lesser with p3. So we can say p1 is greater than p2 greater than p3. p1 greater than p2 greater than p3 is the option number 2, right? Let's go with the next question. Okay. So 37, a parallel plate capacitor is charged by connecting it to a battery through a resistor. If I is the current in the circuit, then in the gap between the plates, kya hoga? So I hope all of you have went through this chapter called electromagnetic waves, where when you talk about these things, that the current flowing through this is said to be IC, and because the current is flowing here, the charge is changing here. The charge is changing here, which is creating electric field here. And in case you keep a magnetic needle here, it will go for a deflection. We call it, obviously whenever there is a magnetic field being produced, there is a current responsible. We call this current, in this case here, as displacement current, and the current flowing in the wire is named as conducting current, right? So conducting current is equals to the displacement current, right? IC is equals to ID. So in whichever direction it's going, the same direction ID is going. They follow the same direction. They follow KCL then. So displacement current of magnitude equal to I flow in the same direction as I, right? Opposite nahi hoga, the same direction, hai na? Greater than I hoga nahi. There is no current, will not be the option. Displacement current of the magnitude equal to the I flow in the same direction as of the conducting current. Yes. 38. The property which is not an electromagnetic wave traveling in the free space is that. Okay, let's go through the it's a theoretical question, dear all. It's a theoretical question. When you talk about theoretical question, you have to know the basic concepts and basic theory about the topic. Electromagnetic wave traveling in the free space is that they travel with, this is the right statement. They originate from charge moving with, actually they originate from charge of an accelerated charge. When they oscillate, they actually produce EM wave. So this is a wrong statement actually. This is a wrong statement. So the question, which of an electron wave traveling in free space is not an property. So this is not a property. EM wave is generated by accelerated charge or maybe oscillating charge. Either of the statement will be right. But this is not right statement. So they originate from a charge moving with uniform speed is a wrong statement. So this is the answer because they are asking not of an electromagnetic wave traveling in the free space. They are transverse in nature. Yes, EM wave is transverse in nature. Energy density in electric field is equal to energy density in magnetic field. Yes, it's also a right option. Half epsilon E square equals to B square by 2 mu naught. Going for the next question, it's a very interesting question to discuss on. Choose the correct circuit which can achieve the bridge balance. So, dear all, generally we think of it as a Wheatstone bridge. So, 10 by 15 is like 2 by 3, and we should have 2 by 3 here also, but what we are getting here is 10 by 15, it's 2 by 1. So, it is not a Wheatstone bridge. Achha, if it's short circuited, like what generally a diode does, it's like a short circuit when it's an ideal diode. Okay? When it's an ideal diode. But by the way, if this diode has some resistance in realistic world, diode will have some resistance. If the diode has some resistance, then this parallel combination will have a resistance lesser than 5 ohm at least. So they will not go for the ratio of 2 by 3 to 2 by 3 anyway. So option A is surely wrong. Let's go for the next option. When I say balance bridge, matlab the value of galvanometric current, the current through galvanometer should be 0. The reading of G should be 0, right? So going forward, 10 by 15 and here 10 by 5, right? Let's come back, let's see more question. 10 by 15 plus 10 by 5 with this diode. This diode might have some resistance because they have said which can achieve matlab probability. Matlab we are talking about the probability, maybe type things. If there's a maybe type things, this diode can have some resistance, right? This diode can have some resistance, right? The problem with this circuit and compared to this circuit is, this circuit has a positive polarity here. This circuit has a positive polarity here, which means it's already reversed bias. So current cannot flow through this. When current cannot flow through this, they cannot be in the same ratio. Whereas this has positive polarity here, minus here, and this diode may have a resistance equals to 10, when then 10 plus 5 will become 15. So 10 by 15 will be 2:3, and this 10 by 15 also will be 2:3. This circuit, this case can be a case where they will achieve a balanced condition of the circuit, which we call as balanced Wheatstone bridge. It might be a case. I'm saying the problem with this is it's a reverse bias, so no current will flow, so then the ratio won't be maintained. As we know, if it's a Wheatstone bridge, this ratio R1 by R2 is equals to R3 by R4 is to be maintained, right? If I name it like this, this is the condition for a balanced Wheatstone bridge. R1 if I mention this, and R2 I mention this, R3 I mention this, R4 I mention this, they should be proportionate for this circuit to be balanced. But the current flowing through galvanometer should be 0 to achieve the balanced condition, right? So considering all this, again it's a reverse bias, so no current will flow through this. So option 4 is anyways wrong, 2 is wrong. 1 cannot achieve, we have seen. So after seeing through all this, obviously 4 is the perfect answer, right? Going with the next question. Okay, 40 number. If the plates of parallel plate capacitor and connected 1A battery are moved close to each other, if the plates of a parallel plate capacitor connected through a battery are moved close to each other, a constant battery, 1A battery hona chahiye. This must be a battery only. So if there is a capacitor plate with a battery here, this question might be talking about that there is a battery been plugged in which is not being removed, and the plates are coming close to each other. The plates are being brought close to each other. So when the plates are being brought close to each other, the D is reducing, which implies that C is equals to epsilon naught A by D. So when D decreases, C increases, which implies that when C actually increases, for a constant given battery, Q is equals to CV, right? Q is equals to CV. So when V is given constant and C is increasing, implies Q is also increasing. So option A is the right. The energy stored in it decreases. No, actually energy stored formula will be half CV square. If V is constant, C is increasing, energy should increase. And they have mentioned decreases, so wrong option. C, it's a capacitance increases. Obviously C is increasing as D is decreasing. The ratio of charge to its potential remains same. Ratio of charge to potential. Charge to potential. And what is charge? Charge is CV by V, right? Q is equals to CV, right? Q is equals to CV, right? Which means the ratio of charge to potential means C only, right? Whereas C is increasing. So D is a wrong statement. The product of charge and V increases. Charge and V. What is charge? Charge is CV into V, which is CV square. Maybe V is constant, but C is increasing, right? C is increasing. We have seen this. C is increasing. So, increases. Choose the most appropriate answer. A, C, and E is right. We'll go for option number 4. We'll go for option number 4. Let's go for the next question. 41. A force defined by F equals to alpha T square plus beta T acts on a particle at a given time. The factor which is dimensionless if alpha and beta are constant. So dear all, if alpha is equals to or F is equals to alpha T square plus beta T, so the dimension of this, and dimension of this must be same, right? Because by principle of homogeneity, the same dimensions are added and is equaled, right? Principle of homogeneity. So going back there, alpha into T square, if I compare this with force. You can go with the unit of force or you can go with the dimension of force. If in case people don't know the dimension of force, the dimension of force is M into A. M is M, LT minus 2. So coming back there, this is like MLT minus 2. So alpha dimensionally will become, we should use the third bracket to make it look better, T minus 4. Alpha is MLT minus 4. Going for the next, what is beta? So beta into T has the same dimension as, force again, because this plus this plus this, if things are getting summed up or being equaled, they must be having the same dimension, right? So beta into T is equals to M L T minus 2, MLT minus 2, MA, A is LT minus 2. So beta into T is MLT minus 2. So beta is equals to, dimensionally, beta is dimensionally MLT minus 2 by T, MLT minus 3. So now what combination will give you a factor dimensionless, right? We have MLT minus 4 here, MLT minus 3 here. If I divide both of them and multiply alpha with T, right? MLT minus 4 by MLT minus 3, ML T minus 3, T minus 1 will be there. So if I multiply by a T here, you will get dimensionless. That T will be T minus 1, T plus 1. That will be dimensionless, right? So what is it actually? Alpha. What is it actually? Beta. Alpha by beta into T. Alpha by beta into T is the answer. Yes or no? Alpha by beta, alpha T by beta is the answer, right? Going forward with the next question. 42. A metallic bar of Young's modulus 0.5 10 to the power 11 Newton per meter square and the coefficient of thermal expansion 10 to the power 5 minus 5 per degree Celsius, length 1 meter and the area of cross section 3 meters square is heated from 0 to 100 degrees Celsius without expansion on bending. Okay. The compressive force developed in it is F is equals to YA alpha del theta. YA alpha del theta is the formula for this type of question where this compressive force. It's a very much derived formula. If you put the value of Y, the value of Y is 0.5 10 to the power 11 What is the value of A? The value of A is 10 to the power minus 3. The value of alpha 10 to the power minus 5. The value of del theta is 100. When you solve this question, when you solve this all, after solving this, you'll get it as 50, 10 to the power 3 newtons, because the force. So unit will be force, right? Newton. Going with the next question, 43. A small telescope has an objective of focal length 140 centimeter and eyepiece of focal length 5 centimeter. The magnifying power of the telescope for viewing a distant object. So the formula for the magnifying power here is FO by FE for image forming at infinity. For image forming at infinity, FO by FE bracket में 1 plus FE by D for image forming at some distance D, least distance of distinct vision, whereas for image forming at infinity, FO by FE, which comes out to be 140 by 5, which you solve and get 28. So option number 4, right? 44. An iron bar of length L has magnetic moment M. It is bent at the middle of its length such that the two arms makes angle 60 degree with each other, right? So let's say this was the bar, north pole here and south pole there, like this. You bent it, becomes north and south now. This is 60 degree. From the middle, matlab this length is L by 2. This length is also L by 2. We all know magnetic dipole moment is M into L by 2, right? So what is the effective magnetic dipole moment is this length. This is the M effective, right? Now, what is the initial dipole moment? Initial dipole moment was M is equals to M into L. Now this is L by 2. M into L by 2, which becomes M by 2. M into L by 2, which becomes M by 2. 60 here, same length, this is also 60. This is also 60. So this is also the same length because this becomes an equilateral triangle. So the length here is also L by 2. So the value of this will be also M by 2, right? Because magnetic dipole moment is M into L by 2. So it's again M by 2, right? So the answer is option 4, right? Going with the next question, 45. A 10 microfarad capacitor is connected to a 210 volt. So voltage, when they don't mention anything, it's VRMS. As shown in figure, the peak current. So what is the peak current formula then? IRMS is VRMS by Z in general. In this case, it's a pure capacitive circuit. It's a pure capacitive circuit. So we'll mention it as XC, VRMS by XC. What is the value of VRMS? 210. By the way, what is the value of I peak? They want to find I peak. So what is the value of I peak? I peak is IRMS into root 2, right? Because I peak by root 2 is IRMS. So whatever it is, 210 by XC. What is XC? 1 by omega C. The value of 1 by omega C. The value of omega is 2 pi F. XC, 1 by omega C. XC, C value is what? 10 into 10 to the power minus 6, right? To make this as peak value, to make this as peak value, we'll replace it, to make this as peak value, we'll replace by V peak. So V peak is 210 into root 2. As I peak is IRMS root 2, V peak is also VRMS into root 2, right? So 210 root 2 by 1 by 2 pi F, which is omega, into XC. When you solve this, this comes out to be 0.93 ampere. I'm not going for the calculation right now. I assume we can do this calculation. Simply this will come out to be option number 4. We can just put the value of pi as 3.14 and solve it, right? Okay, let's go for the next question. Two heaters A and B have power rating 1 kilowatt and 2 kilowatt respectively. Those two are first rented in series and then in parallel. First of all, when they said as two heaters of power 1 kilowatt and 2 kilowatt. When nothing is mentioned about the rating voltages, we'll write it as the same voltage, right? So PA by PB, the formula for P is equal to V square by R. So we'll assume the V is same, V square by RA by V square by RB, V square cancel. We can write this as RB by RA, right? We can write this as RB by RA. What is the value of PA by PB? The value of PA by PB is 1 by 2. Okay, so the value of RB could be written as some R, and the value of RA could be written as some 2R, where R is some constant used to make it like this, right? Those two are first connected in series. So when they are connected in series, that's R plus 2R will become 3R. When they are connected in parallel, this R into 2R by R plus 2R, right? R1, R2 by R1 plus R2, which becomes 2R square by 3R. R cancel, becomes 2R by 3, right? That's the R series, that's the R parallel. So then in parallel to a fixed power source, the ratio, fixed power source is the same voltage, right? The ratio of power output for these two cases is power for series to power for parallel is V square by R for series by V square for R for parallel. V square cancel, which becomes R parallel by R series. What is the value of R parallel? 2R by 3. What is the value of R series? 3R. R cancel, becomes 2 by 9, 3 into 9, right? That becomes option number 4 is the answer for this, right? Let's go with the question number 47. Now, it's a VT graph given. Dear all, the slope of VT graph is a dV by dt, which becomes equals to acceleration, right? So let's try to draw the graph of acceleration. We can see the slope here is constant, slope here is 0, and slope here is negative, right? So acceleration here is positive, like slope matlab, the angle here is acute, so the slope here is positive. Slope here is 0 because it's parallel to the x-axis. Slope here is negative because the angle formed here is obtuse, right? So acceleration is positive, 0 and negative. Option number 1 will be the perfect match for this condition, right? We have to draw the corresponding AT graph. Going with the next question, 48. If the mass of the bob in a simple pendulum is increased to thrice its original mass and its length is made half of its original length, then the new time period of the oscillation is x by 2. If I assume the initial time period is x, this is x by 2, right? That's what they mean. Then the value of x is. Coming back, the formula for simple pendulum, 2 pi root over L by G, right? So since we are discussing about simple pendulum, we will not discuss about the mass. So there is no dependency of mass actually, right? So 2 pi root over L by G, where the length is made half of its original. If this was the time which was mentioned as x, now the new time is mentioned as 2 pi root over L by 2G, which could be written as T by root 2 then, right? 2 pi root over L by G, root 2 taken out, T by root, right? Where is the value of new time period is x by 2. New time period is x by 2. Okay. New time period is x by 2. So we'll not compare with this because the new time period has been named as x by 2. है ना? नए वाले time period को we have mentioned as x by 2. So this is mentioned as x by 2. Okay. So what is it? T by root 2. So if I compare this with x by 2, right? Then the value of x, after solving this will come out to be root 2. I guess there is no option given. The value of x is coming out to be root 2. So might be this should be root 2. There's a mistake in the options here. There must be root 2 as an option, right? If the mass of the bob in simple pendulum is increased to thrice its original mass, so mass is not dependent. Length is half its original length. So L by 2, okay? So new time period is T by root 2, right? And the new time period is of oscillation is x by 2 times its original. So if I mention this as x, this is x by 2 times, right? Then the value of x, they haven't said about x, so x must be having the value of root 2. The value of x must be root 2, right? The value of x must be root 2. Is it okay to all? If I put the value here as x, so this becomes 2 by root 2, which is root 2. So the answer should be root 2, and there is no option like this. I guess option 4 should be written as root 2. Okay? There's a mistake here. Please correct this. 49. So the minimum energy required to launch a satellite of mass m from the surface of the Earth of mass m and radius r in a circular orbit. So what this mentions like is, this is like there. You are launching here. So when particle is on the Earth, the energy here is initial energy, which is minus of gmm by r, right? Now you're putting some energy here. It finally reaches to some height, and now it goes to circular orbit here. Somebody here must be having some energy. So on the orbit, the energy formula is minus gmm by 2r. So value of r here is the distance from the center. This is 2r given, right? In a circular orbit of radius, altitude 2r. Altitude 2r, this is 2r. So 2r plus r, 3r. So this is minus gmm by 6r, right? Minus gmm by 6r. Here it is minus gmm by r. So how much energy we have put in here? We have to find this. So E plus of minus gmm by r is equals to minus of gmm by 6r, which goes that side and becomes, taking out this gmm by r common, 1 minus 1 by 6 becomes 5 gmm, 5 gmm, 5 gmm by 6r. So the option will be option number 3, right? Let's go with the last question of the day. A sheet is placed on a horizontal surface in front of, this is a sheet, right? For strong magnetic pole, let's say there is a magnetic pole. Okay. There's a magnetic pole here. There's a magnetic pole here. A force is needed to hold the sheet. A force is needed to, let's read the option. A force is needed to hold the sheet there if it is magnetic. If it is magnetic, but the sheet is magnetic, implies this will be magnetized. So chance of this this sheet getting attracted towards the poles, right? So yes, we have to give a force to hold it. So option A is right. Option B, hold the sheet there if it is non-magnetic. If it is non-magnetic, we don't need any force. It will be there. See, move the sheet away from the pole with uniform velocity if it is conducting. Move the sheet away from the, if it is conducting and you're moving it away, if you're moving this sheet away, there'll be a flow of eddy current actually. I mean, you can think like there'll be a rate of flow, there'll be a rate of change of flux here. There'll be a rate of change of flux resulting a flow of current. And when you are trying to move it, the sheet will try to obstruct this motion by Faraday law. By Faraday law. Faraday says that by direction of induced change such so as to oppose the cause. So if you're trying to move it down, the force will be developed such that it is opposing this motion, right? It's opposing the motion. So option C is good, right? We require the force to move it. Move the sheet away from the pole. When you're trying to move it, you have to put a force, otherwise it cannot move. Option D, move the sheet away from the pole with uniform velocity if it is both non-conducting. Okay, if it is non-conducting, there's no eddy current, there's no force required, number one. And non-polar, it won't be magnetized. So again, no force required, right? Choose the correct statement from the option. So I can see A and C is the perfect one. D is wrong because when it's non-conducting, there's no eddy current, there's no Faraday law logic going on. So we understand it won't be obstructed in the motion. And same for non-polar. So together implies that there is no problem in moving this sheet from here. So A and C, I feel is the best option to go for. So option number 4 is the right answer so far, right? So this brings me to the completion of the discussion. So this brings me to the end of the discussion of the NEET paper 2024. I hope all of you have liked it. Thank you so much for joining the session and attending the session. We'll be there to help you in case if there's any other discussion to be discussed. You can put your comment down. Thank you so much. Stay put. Keep studying, keep growing, go forward. Thank you so much. Thank you so much.